Good morning, everybody, and welcome to Workshop 3 for our Australasian Seed Science Conference. I'm Lydia Guia. I'm from the National Seed Bank at the Australian National Botanic Gardens, one of the hosts of the conference. And I want to just start this morning by acknowledging the traditional owners, the Ngunnawal and Ngambri people, who are the traditional custodians of the land that I'm meeting from. But I also welcome all of you to please start to introduce yourselves in the chat. Let us know who you are and the country that you're coming to us from, and anything you would like to say about stats or what you're interested in learning about today. So I'm only going to talk to you for a few minutes just to give us a bit of an introduction and go through some of the housekeeping for today. And just to let you know, the program, which hopefully you can see on the screen right now, we're going to run through a mix of lectures and hands-on sessions where you'll be running code yourself. So we'll have a few lectures this morning, then a little bit of setup ready to go in R, a break for a short time, then a session on data wrangling and data visualization, a break for lunch where feel free to join the Zoom and we will set up smaller breakout rooms so that you can chat and get to know each other and talk with other participants and demonstrators from the workshop, or feel free to just take time out if that's what you need to do. Then we'll come back at 1 o'clock and go through linear models, another short break, and then finish off the day with generalized linear models. So this program, Aranex will share in the chat, so you'll all have the program at hand if you need to check that at any point. And also we'll try and put it up on screen throughout the day just so you can keep track of where we are. So I just wanted to start with going through why we wanted to have a seed workshop. And my colleague Emma shared this cartoon recently and it really resonated with me. I looked at it and thought, it really describes my personal experience with seed biology and stats, and that is that I came into seed biology because I really had a deep love of nature and plants and was so curious about seed germination. And it was only a little further into my research career that I realized just how much statistics and R coding was involved in this field, and it's sort of this hidden element to the kind of work that we do. And so we wanted to have a stats workshop because we collectively, among the organising and scientific committees for the conference, we knew that seed science generates a lot of data. That data has a certain structure and certain properties that need appropriate statistical analysis. And I don't know about everybody else on the call, but personally for me in my undergrad training, we got just sort of to the point of stats where you could almost deal with seed data and you learned about ANOVAs and you could analyze certain types of experiments. But I realized as I went through honours and PhD and into my career that it was much more complex than that, and it required specialist training, and a lot of the courses out there that did the kind of modelling that we needed were, even if they were focused on ecology, they didn't use seed examples. And so we really wanted to bring everything together and run this workshop for you. The second part of the workshop is that we're running everything in R and R Studio. So the reason that we chose this is that it's a free and open source software, or they both are free open source softwares. They're very widely used in the research community, and it means that there's a really big support group out there. If you run into a problem, it's very easy to Google that problem or a line of code and find answers that you need to continue your work. R and R Studio allow you to clean your data, analyze your data, graph your data. You can do everything in the one program. And importantly, they give you reproducible workflows, and you can reproduce results for publication and easily arrive at the same answer and follow the process that you use to get there. So to run today's workshop, we have three excellent facilitators, instructors, moderators, whatever we want to call them. And they're all really highly capable, credentialed, well-published authors in their own fields. You can look up their Google Scholar and find papers on this sort of work where they use great stats. And so I won't read through the technical biographies that you see on the screen, but I thought instead I'd give you a bit of my sort of personal take on what it's like working with these people and why they're a perfect fit for this workshop. So Adrienne Nicotra is the one who will start the day off and walk us through some experiment design. And I've had the pleasure of working with Adrienne for the past few years. And one thing I've really admired and learned from Adrienne is right from the stage of thinking about hypotheses and experiment design for answering those hypotheses, Adrienne's always steps ahead and also imagining what the data are going to look like and how we might analyse them and whether the analyses actually would allow us to answer the question that we're dreaming up. So I think it's just an amazing skill that she has to see the whole process, all the data, and is also an excellent teacher and supporter of early career researchers, which is lovely. Adrienne introduced me to Terry, who's also been a phenomenal person to work with in the last couple of years. Terry's a biostatistician, and the thing that I've personally really loved about working with Terry is that she's got all of the statistical knowledge and knows how to do things and why, but she also checks in with us to ask, but does that make biological sense, or is this logical in an ecological framework? And so she's always sort of checking in to make sure that the stats and the ecology are matching up, which is really lovely thing to be able to do. And Terry's also an excellent code writer and very friendly and lovely approach to her code editing and assistance. And so finally, our third presenter today is Dr. Sergei Rospa. I had the pleasure of meeting Sergei a few years ago at a seed ecology conference in Regensburg, where he's based. So Sergei, it's something like the middle of the night for him at the moment. He'll join us later today for his sessions, but he won't be online this morning. But yes, Sergei is a vegetation ecologist. He's presented in the conference. You might have seen his work that way. But one thing that I really regretted about this seed ecology conference in Regensburg was that there was a pre-conference stats workshop run by Sergei and other colleagues, that all I heard for the whole conference was how amazing that workshop had been and how useful people found it. And I was just so regretful that I hadn't signed up myself. So we're really lucky that we were able to drag Sergei in to give us at least some of that workshop in today's session. That one was actually a whole week's worth of stats. We've had to just go with one day. So that is a good place to mention that there's a lot we could have covered, but we do only have one day. So we hope that we've picked the things that are going to be most helpful to people. So that's a bit about our presenters. And the other side of it is all of you, the participants. So last I checked, we had something like 114 people signed up to take part in this workshop, which is fantastic, from 20 countries. And we think that among you, there's probably a very broad range of experience. Some of you might be here because you are getting started in R or getting started in seeds, or some of you might be here because you want to learn some new tricks with your code or different ways to look at your data. And so we really want to make the most of that diverse experience in the room and make it a really joint and welcoming workshop where we all chip in and help each other. So please throw any questions into the chat, but also please feel free to jump in and answer other people's questions if you know the answer to something or have an idea of how you might be able to work through it. Finally, I think that leads to the housekeeping for the day. So I forgot to warn, but I did ask Amy to start recording, that we will record today so that this is available for people, either if they can't join us right now because it's not the right time zone, or if, for example, you feel like you fall a little behind or you want to rewatch something, those recordings will all be available to dip back into later and you can follow along at your own pace. If you don't want to be recorded, please feel free to keep your camera and microphone turned off, and that will keep you out of the recording. But we hope that everyone will be happy to take part. So we have only one Zoom link for the whole day, which is the link that you've used to join. And there's a lot of us obviously joining for the day. So we ask you to please just remember that we're all in the one room. So if you can keep your microphones muted when you're not speaking, that would be great. And I can see a lot of you have already done that. But the other thing is we invite you to please have your cameras on. At the moment, I can see a few people with their cameras on, but many of you are also off screen. One reason we would love you to turn your cameras on right now, if you don't mind, is that we're really keen to have a group photo of the workshop. So I might give everyone a moment to turn their cameras on if they're happy to be in that photo. And then we'll ask Amy if she can take that in a moment. So if everybody's feeling ready, do a countdown and a big smile. So 3, 2, 1. And hopefully you managed to grab that, Amy. Let us know if we need to try again. One thing about having so many people in the room and the cameras on is that you might want to, up the top on your screen in view, just choose to use the speaker view so that you always at least can see the person who's speaking. But that's up to you. So when we hit the brakes, we will ask everyone to just turn off their video microphone at that point, assuming that you're not going to be at your computer. I'll just repeat again about asking questions in the chat. Please, in the chat, please use the chat. Ask any questions you like. We've got a few of us helping out in the background, and a thank you to Rocco and Caroline and Gemma, who are helping keep an eye on the chat or helping chat with you at lunchtime. It's excellent to have people in the background helping out. So yes, if our moderators see that you've got a question that's of broad interest, we might pass that on to the presenter to talk about with the whole room. If it's something a bit technical or coding related, we'll pop you out into a breakout room with somebody who can help and look at your screen and help fix that coding problem. Or at times we might ask for spoken questions, and just because there are a few of us in the room, if you can use the little raise hand button, I think at the bottom of your screen there should be reactions. I'm not seeing that on mine, but hopefully you guys have that on yours. If not, feel free to just unmute and wave around or something, whatever works for you. And yes, please do jump in, answer other people's questions. The last thing that I'll finish on is that the homework was essential, and I really hope that everybody's managed to do it. If you have done your homework, you should have downloaded and installed RStudio, the latest versions of both, and you had a link to a Dropbox folder which looked like this screenshot on the right. There were a couple of files in there, a data file and 3 scripts, and hopefully you can run those scripts, and if you see warning messages, that's fine, but if you see error messages, we'll need to help you out with some troubleshooting. So it's a good time. If you have had any troubles and haven't had them fixed yet, please chuck them into the chat and we'll try and get that started. If you haven't done the homework, it is okay, don't leave. You can sit back and watch, but we do really think you'll get more out of it if you can follow along. So Adrienne has kindly said she would be happy for people to duck out and be downloading these things during her next presentation. It's just a presentation rather than a hands-on session, so hopefully you can be listening and also downloading the files in the background. And that's it from me. This is just another recap of the program, and to hand over to Adrienne to take us on to part 1, an introduction to robust experimental design.
Thanks, Lydia, and lovely to see everyone this morning. I'm not losing my voice, don't worry. Yeah, as Lydia said, if anyone didn't get a chance to download and do the homework, please go ahead and do that in the background. And if you've had any problems, have any issues with it not working, just pop something in the text box and one of our helpers will try to help you out. I am going to share my screen now. All right, hopefully you can all see that. Thumbs up if you can. Excellent. Yeah, so as Lydia said, feel free to have your cameras on and raise your hand if you have a question, or put it in the chat box. And my question is, just give me a second to get my act together here. I'm just trying to find the chat box. I seem to have lost it. Okay, team, I don't have the chat box, but that's okay because I can't really answer things in the chat box anyway. I'll leave that to my co-hosts. It wouldn't be a normal Zoom chat if something did not work, right? Okay, so as Lydia said, the aim in this first session is kind of to make sure that we're all on the same page, to outline the objectives of the workshop, and to get you all set up for the more R-focused elements to come. I wanted to just take a little bit of time and encourage you, as I'm talking, to reflect on your own work, but also just to think back on the amazing work that we've heard about this week. I have to say that listening to those talks, and I had the pleasure of sharing the first session on Monday morning, I just came away thinking this is an incredibly exciting time to be doing seed science. We heard about projects from all over the world. I've noticed that, you know, there are people from all over the world on the workshop today. And we've heard about major international collaborations, people working with each other from lots of different places, drawing together these beautiful data sets, drawing together long-term data sets. And we've also heard about the impact that seed science is having, and that's just so exciting. And clearly one reason for that is the technology and all of that. But I think it's also in part that we have more interesting and more powerful analyses that are giving us the tools to really probe our data and answer some exciting questions. So I guess my take is that the stats that we'll learn about today, the stats are only as good as the data that go into them, right? So before you start grappling with how to analyze the data, I thought we could have a quick think about the sorts of data that we collect, and how we design our experiments to ensure that we get data of the best quality out of them. So apologies to those of you who are aficionados. I'm going to start at square one, and that way we sort of all have the same foundation to work from. Cool. So I'm going to start by introducing myself, and confessing that I got into seed ecology probably about a dozen years ago, when I started a collaboration with our lovely colleagues at the ANBG. And up till then, my focus was reproductive ecology and ecophysiology, and I was interested in fruit and seed production, but I honestly hadn't thought about what makes them germinate at all. And so that was a wonderful change in my sort of research portfolio, and I've been doing seed ecology amongst other things ever since. So I'm not a seed scientist by origin. I'm also not a statistician at all. In fact, my R skills reflect my generation and are fairly rudimentary. Luckily, I've got really clever collaborators and students. And I do make a point to collaborate with statisticians like Terry whenever I can. And I love keeping up on the challenging and evolving ideas in stats and how we analyze our data. But the reason I'm here is I've always been really passionate about experimental design. I find it really fun. And I love the power and the insight that we can gain from statistics with a well-designed experiment. So just for context, my group is interested in the things you can see on the slide, how climate change will affect alpine plant reproductive ecology is a really big one for us. And we look at seed production and seed and seedling characteristics as part of that. I'm also interested in the evolution of plant responses to water availability and temperature stress, phenotypic plasticity and ontogeny. And I'm interested in phenotypic evolution. And the picture here is one of my research assistants. It's actually an old picture of my daughter. She was 2 at the time, and I think she'd spent maybe 1 too many holidays out doing plant science, because she's painting with pollen here. Unfortunately, pelargoniums aren't really very good at selfing, so I don't think this experiment was successful. But I put that there just to say, if anyone ever wants to talk about the joys of doing science with wonderful family, I'm always keen to do that as well. Because yes, my children have been dragged around the world doing field biology. All right. Now we're just going to do a little bit of polling to learn a little bit about you guys. So when I ask the question, what does your ideal model look like? What's the first thing that pops into your head? The one on the left, a guide to model railways, or the one on the right? Obviously, this is a continuum. So, Amy, if you can launch the poll, that would be great. And can everyone answer this question on a scale of 1 to 10, where 1 is the model train set and 10 is that beautiful Greek object there? What does the word model mean to you? Now, don't everyone pick 5 or 6. That's cheap. Give it a few more minutes. Not everyone's had a chance to answer yet. And I like that there's someone who, you know, there's a few people who admit that when I say model, they go straight to Greece. That's brilliant. All right, that seems to be pretty much everyone, I think. Amy, do you want to share our results? So if you take a look at that, what you'll see is that those of us here today, we run the full gamut. For folks for whom that sort of statistical lingo is a bit of an anathema, and certainly not the first thing that comes to mind, to people for whom a model is a statistical model, and that's the first thing that comes to mind. And we're really excited to have everyone here, and we want everyone to feel welcome, whatever your engagement has been up to this point in stats. As Lydia said, we grappled with what we should cover, and what we've tried to do is cover quite a lot. And because it's recorded, you've always got the chance to go back and listen to things again if you missed something. So thank you for tolerating my little icebreaker there. Oh, okay. What's this? You can answer in the chat box, but I can't see the chat box, so somebody will have to shout it out at some point. Anyone?
They look like railroad tracks, Adrienne.
I know they're railroad tracks, but they're linear and nonlinear models. Okay, and I'm just checking to see if you're awake. All right, so why did I do that to you, the poll? I mean, not the dumb joke. Just to highlight where we all come from, and to make that point that everyone's welcome, and we encourage you to engage with the material in whatever way works for you. So I guess my starting point when it comes to doing an experiment is that it's always really important to consider what your question is, what your hypothesis is, and where you want to go from the outset. This is a picture from the Hangduan Mountains, which is sort of the foothills of the Himalaya, where I was doing reproductive ecology with a student on Meconopsis poppies some years ago. So what I want you to think about is having an approach that always says, what's my question? What's my hypothesis? What's the design that's going to most powerfully help me to test that hypothesis and answer my question? And so if you're letting your question drive your design, there's a few things that you really want to think deeply about before you set off. And I'll touch on a few of these today: replication, blocking or nesting, and the notorious pseudoreplication. And I suppose part of what I want you to think about is how experimental design helps you when you know fully well that you can't control everything. There's always noise, there's always sources of error, there's always variance that we cannot account for. How do you best design an experiment so that your signal rises from that noise? And the other thing that I'll end on is a plea that you get to know your data before you subject it to statistics, which is where I will end up leaving off and passing to Terry. So I'll give you the take-home messages before I go any further. What I'm really going to be pushing with you today is that I always want you to make the most of your limited time and your space and your resources. Your time is really valuable. So make sure that when you do an experiment and you measure things, you're measuring them for a really good reason, not just because you can, but because you have a question about them and you think those data will help you answer the question. Never forget your time is valuable. I also want you to leave here feeling more confident about when to replicate and where to put your replicates. And I want you to start thinking about interactions and get more comfortable interpreting the effects of interacting factors, which so often come into our seed ecology designs. So let's start just big picture. What does modern seed ecology look like, or modern seed science look like? Looking at the room and thinking about the talks that we've heard over the last few days, we know that seed science ranges from field studies on naturally occurring plants, where people are going out and asking questions about reproductive allocation and seed production. They're asking questions about how resource availability or disturbance history or community composition are affecting fruit and seed production and the traits of those seeds. They're asking about the soil seed bank, what's in the soil seed bank, what drives germination, what drives recruitment. Field studies are wonderful. I love field studies. They're exciting because they're really realistic. And as you all know, they're challenging because there's very, very little control. We've also heard about a lot of really cool seed science that's going on in sort of what we would call common garden settings, where we're looking at how different levels or combinations of resources and conditions are affecting things like emergence and recruitment. And by common garden, I mean when you go out and you introduce seeds or you introduce seedlings into an environment, you control the conditions, you run the design, you set up your design, you run the experiment, all in a sort of contained place. They're not naturally recruiting individuals. And you can look at things like effects of competition, or you can bring together provenances of a particular species, grow them under common conditions in a semi-natural setting. So it's a semi-controlled environment. It gives you realistic environmental fluctuations and variation, but you've got your artificially placed research subjects so you can compare them under common conditions. And those sorts of experiments can also be really powerful. And then finally, we've got our sort of workhorse of our glass house, our growth chamber, our incubator studies, which are really powerful for looking at things like what are the germination cues that drive germination for a given species, what are the mechanisms underlying patterns of seed production, and how do things affect the characteristics of our seeds at a mechanistic level. So for example, if you're interested in looking at, say, hormone levels and how they affect germination, or doing an experiment where you provide different sorts of hormones or different sorts of temperatures, you can't get better than a controlled growth chamber incubator experiment. And you're making a choice then, right? You are sacrificing realism for control. You've got the opportunity to manipulate, to monitor. You've got access much easier than if something's in the field. But when we're doing experiments in these settings, we have to ask ourselves, how do we make the conditions as biologically relevant as possible? And that's always a challenge. For me, I love to try and bring these things together, to use glasshouses to gain insight about mechanism, to use common garden experiments to tease apart differences, say among species or provenances, and then to go out and see if I can see those patterns playing out in the real world. So I'm not advocating one method over another. The other sort of seed science I think is really important to keep in mind is that we're often dealing with legacy data. So germination trials that have been done over years, potentially with different methods, or we're doing research on old and really valuable seeds. I love this picture. I got it off the Q webpage. I think this is what lots of people think a seed bank looks like, and I think it's really cute, all those little jars. It's more like my spice cupboard than the seed bank, but anyway. I think it's really important when we think about that sort of legacy data and these old seeds to remember that we design within constraints. So if you're going to do an experiment where you're using really valuable seed, you don't want to design that uses a kilogram of seed, right? You want to come up with something really elegant, powerful, but elegant, that respects the value of that resource. So given that we can't control everything and that we want to be able to do field work and common garden experiments, design becomes really critical. Because even when you are trying to control everything, right, there is always error and there is always noise. This is a picture of a really gorgeous, beautiful, state-of-the-art climate-controlled glasshouse at ANU. What could possibly go wrong? Those of you who live in Canberra know that this is a picture of the hailstorm we had January 2020. And that's me standing in the semi-repaired glass house a few months later. The roof still had holes in it. No amount of experimental design could have prevented that, obviously, but that's a pretty unusual circumstance. So we'll chat a bit more now about design in the context of seed germination data. I found the chat. So exciting. Okay.
Adrienne, there's a question on the chat.
Is there? Okay, cool.
Yes, so Ella asks, what did you do in that instance? Did you have to start again?
Ella, it was absolutely heartbreaking. Yes, we had an experiment running. We had been doing controlled crosses with an alpine plant where we'd been crossing different lines to try and look at plasticity and phenology and germination and how growth conditions affected those things and whether there were transgenerational effects. We were on the 3rd generation and 3 months from the end of the experiment, and we lost the experiment. So there was lots of data that we'd collected up to that point, but we didn't manage to harvest the plants and get the seeds. So it was heartbreaking. As Rocco, who's here today as a helper, can tell you, that was supposed to be one of his big PhD experiments. So. But we're resourceful and we come up with other experiments to step in and fill the gap, right, Rocco? Yeah, okay. So we're looking at some seed germination data here. And I'll ask you to think a little bit about what's special about seed germination data. we think seeds are special because we all love them, because here we are at the Seed Science Conference. But what is it? What are some of the distinctive elements of data when you're dealing with seeds? Answer in the chat if you like, and have a look at that little data set while you do.
Could you ask the question again? Sorry.
What's distinctive about seed data? So Jose's posted something which is one of the biggest ones, right? Seed germination is binary. You do it or you don't. You don't get to half germinate. And that means that the data are need to be handled in a slightly different way to something like growth rate or the mass of the seed. Yeah, viability becomes a really interesting one. A seed's not a seed. We've sort of agreed as a community that if a seed is inviable, it shouldn't be treated in the same way as a viable seed. There should be an assessment of viability separate from an assessment of what the characteristics of the viable seeds are like. The census structure is something Caroline has said. Caroline, do you mind unmuting and explaining what you mean by that?
Sorry, it's a time, it has a temporal element to it.
Yeah.
So we're, taking data.
Sorry,
Yeah.
go ahead.
Yeah, multiple pieces of data over time.
Yeah. So that means that you can be collecting repeated measures, right? And that your question could be specifically about time. Yeah. We might have a number of provinces that all get to the same total germination, but they do it at a different rate, a different pattern, and that might be your question. Dormancy is a really fascinating question. Do you want to alleviate dormancy? Do you not want to alleviate dormancy before you do an experiment? It's a really good one to think about. And you know, how many seeds do you put in a dish? Do you look at seeds as a population, or can you look at them as individuals? And Ellis pointed out, you know, that you can affect what happens in your dishes so much just by the conditions under which you set up the dishes, right? So we're always needing to think about that sort of thing. So yeah, these are the things that make seed data fun and interesting, and bring in some different elements that, you know, might not be the case if you're going out and measuring leaf size or leaf temperature or photosynthesis, which are some of the other things I enjoy doing. So have a look at that data set there. Gemma, this might look familiar to you. These are some of Gemma's early data from one of our collaborations. What I'd like you to do is zoom in on the data set itself and tell me what you think our question was. You can pop it in the chat box if you like. What do you think we were trying to figure out here? Yeah, so from what you can see there, we're looking at the effect of light on germination. You can see we've done a cut test there to deal with the viability question, because we don't want to estimate final percent germination taking into account seeds that were never viable to begin with. So this was part of a big study assessing 40 plus species of alpine plants. I've shown you the final germination data for one species. I haven't shown you the time steps along the way. The seeds were incubated over a simulated year, so they were exposed to different seasons, and germination was recorded along the way. So the other questions that we were looking at then in this study was, what is the seasonal timing of the germination? Do the species have dormancy? And if so, what? And what you can see there is that we had a number of dishes and we had them on different shelves in our incubator, and we had 25 seeds in each dish. So how many samples do we have here? What is a sample when we're doing this kind of seed germination in dishes experiment? Is every seed a sample? Jose suggests that a petri dish or a plate is the sample here. Are we comfortable with that concept? A lot of serious nodding going on. Thank you to those of you who has your cameras on. It makes a huge difference. Much easier than talking to a screen of names. Although I appreciate not everyone has the bandwidth, so no worries, that's okay. So yeah, in this case, we are not treating seeds as replicants, right? We're treating seeds in a dish as a population in a sense, and our samples are the dishes. And we've got one dish per shelf. So we have 4 samples here. So sample size is a really challenging question. And I think it's important once you get to the stats end of the deal to remember that biologically significant and statistically significant are not necessarily the same thing. We want to have a sample size that's big enough to give you the power to detect any statistically significant differences that are out there. But you always need to look at those data and ask yourself, do I think it's biologically meaningful? So for example, you might do an experiment in which you find that percent germination of one population is 98%, and percent germination of another population is 92%, and the data are incredibly clean, and the error is really tight, and they are significantly different. The 98 is higher than the 92. As a seed biologist who's going to do restoration work or who's thinking about conservation, does that mean that you chuck out the 92% because it has lower viability? No, I wouldn't. Seeds are really valuable. It just means that you know that population is slightly lower. But in terms of its utility, say for a conservation point of view, it might be just as good and just as valuable, whereas the population that had 20%, well, we'd think differently about that. So always remember that your stats are a tool, and that you want to design an experiment that allows you to detect significantly different effects, but you never want to forget that you're a biologist first and foremost, and that you want to interpret those data as such. So when trying to decide on sample size, there are really lovely statistical ways to do that. There are power analyses that help you explore the relationships between variance and sample size and error. And power analysis is something that it asks the question, how large does the difference in the means and variance of your populations have to be in order for you to detect it with a given sample size? Or how large a sample size do you have to detect a small difference? And so those statistical approaches are really useful. But there are other things to take into consideration when you're making decisions about sample size. Sample size is always going to be limited by resources being time, being money, being space, being seeds. And you want a design that sort of optimizes the trade-off between really big sample size and the ability to look at the effects of interest, the experimental levels or treatments. I think when you go out to design an experiment, one of the first things that's worth doing is to talk to other people, like the folks on this chat, and people in the conference, and your supervisors and your colleagues, and ask them what their rule of thumb is. You can use published papers as well to assess conventions, and then you can use your own judgment to get a sense of what is a normal sort of variance around data of this sort. And that can be a really important way to support your power analyses and your statistical approaches, right? come back to that biology. And there's a comment here about the soil seed bank, and yeah, soil seed banks are a killer, because you never know what's going to come up, and you're making decisions about do you maximize spatial sampling to represent the community that you're sampling from, but then you've got lots and lots and lots of dishes in your glass house, or do you bring them all back and homogenize them and spread them out, and then you can better understand the impact of the conditions in the glass house, or perhaps you're looking at temperature effects on emergence and that sort of thing. So soil seed banks is a really good example of that sort of mix of fields and controlled conditions, where you do have quite a lot of power to design, but you've got a lot of trade-offs to balance, and you have to carry tons of soil, which is always fun. All right, so the conclusion then was, in that design, there were these 4 samples. We spread them across the 4 shelves of the incubator. We had lots of species, so we were replicating at the species level. And because we were replicating at the species level, we weren't concerned about only having the 4 dishes per species, because our question was about how the species differed. We didn't treat the seeds as replicates, we treated them as a population within the dish. And we looked at percent germination, which then can be analyzed, as we've said earlier, as a binary trait, a trait that either the seed does or doesn't germinate. You come across this term pseudoreplication. I feel like this was a term that was hugely popular with reviewers when I was doing my PhD. It's sort of lost favor now, but it was sort of the death knell on many experiments, where you do an experiment in a given glass house or a given chamber, and the reviewers would say, you've pseudo-replicated, you only had one glass house. So the issue of pseudo-replication is about independence of points. I'll show you a picture of the Petri dish, because pseudo-replication would be a way to describe why we don't treat each of those seeds as an individual replicate. Right, because they're all sitting there together in that dish. They're not independent. They're really important though, right? They're really important to estimating percent germination. I suspect that none of you would set this experiment up with 1 seed in a dish, because that optimizes the independence of your seeds. It's really hard to estimate percent germination with a population of 1. So you're making a decision when you're putting seeds in a dish as to how many seeds do I need in that dish to accurately estimate the variance around the germination. But you need to remember that they're not independent. The other place where this concept of pseudo, hang on. This is my brief digression on independence. We want our points to be independent because our statistical tests are based on certain assumptions: normality, balanced design, and independence. Now, tests vary in how robust they are to violating those assumptions. You can go a long way with data that aren't normally distributed by changing your model or transforming your data. You can go a long way with balanced and unbalanced designs these days. But it's really hard to get around data that are not independent. So yeah, the other time that pseudo-replication becomes an issue that we face in doing seed experiments is chambers. So it's often the case that you have a limited number of growth cabinets or incubators, and you want to set each one at a given temperature, say, or a different sort of diurnal temperature regime or light regime. The problem then is, how do you know that the effect that you're getting when you analyze your data, how do you know that that effect is not just that the chamber on the left side of the room was different to the chamber on the right side of the room? Is it the temperature that's affecting your data, or is it some element of the chamber? And this is really hard to get around. And I'm sure that a lot of you have already thought about these sorts of things. So I'd be curious to hear from a few of you. What do you do to avoid criticism or worries about chamber effects or pseudoreplication in terms of applying temperature or light treatments in particular, but you might have other cases. Anyone have a story they'd like to tell us about this issue? No. Cool. All right. Yeah, blocks and randomization, Vanessa says. Indeed.
I think,
Haven't
Adrienne, it might be worth clarifying about this pseudo-replication, because as you say, there are possibly shelf effects, there are possibly cabinet effects, but it's not clear exactly. Do you mean putting all species of one type on a single shelf, or the light conditions being you having just two growth cabinets, so you just have two light conditions? Is that the kind of what you mean by pseudoreplication?
I guess what I'm thinking about is yeah, cases where I might have 3 incubators and I might be interested in looking for a temperature effect or a light effect. And if I run the experiment with one light treatment in each chamber, I run the risk of the reviewer coming back and telling me that the effect I'm seeing is a chamber effect. And in many cases, you know, it's unavoidable. I only have 3 chambers. So I was, I guess, curious to know if people have run into those sorts of issues and what sort of things we can do to minimize those concerns.
So could you repeat the whole experiment and then just reverse the temperatures in the cabinets?
Yeah, that's the sort of thing I was imagining. So either running it multiple times, or what people sometimes do is they rotate the different treatments through the different chambers. So a given chamber would, over the course of the experiment, subject a set of seeds, a block of seeds, to different temperatures, but you'd be moving the sample so that the chamber effect could be accounted for temporally. But
what about the shelf effect? Is that something that's real?
Yeah, the shelf effect is real. And I'm going there. Should I keep going?
So Ella's asking, is it advisable to move samples within a germination chamber?
Right. Okay. So this is a really good question, and different people deal with it differently. I think part of the reason I have that picture in there of the dishes with the alfoil on them is it's an indication of an experiment where we clearly didn't randomize our wrapped dishes and our unwrapped dishes, right? We've got dishes that we wanted to be subject to fluctuating light levels and dishes that we wanted to be in the dark. If we'd randomized them on the shelf, then the alfoil dishes would have shaded the light dishes and we wouldn't have actually gotten a light effect. So we had to put the light dishes on one side and the alfoil dishes on the other side. And we allowed them to be sort of haphazardly arranged within a shelf, and we moved the light and dark from side to side so that we minimized the side effects, but we didn't move the dishes amongst the shelves. So we kept them consistent and we accounted for the shelf effect in our analysis. And the reason for that was that we suspected, and then we measured to determine that it was true, that there would be a temperature gradient through the chamber. And we wanted to be able to account for that. If we constantly moved everything, we wouldn't know how that temperature gradient was affecting the germination, and we wouldn't actually have had a homogeneous sort of set of conditions that the seeds were exposed to, because some would have been on the warm top shelf early in the experiment, and some would have been on the warm top shelf later in the experiment. So by including a term in our analysis that actually was shelf, we're able to account for that.
Adrienne, Joy had a question. She said, can't you just include chamber as a variable? And if it has a significant effect, then it needs to be considered. And if not, then you haven't shown it had no effect. But I think this was in response to the issue of your completely confounded experiment.
Yeah, so if in that example that I was saying, where I only had the 3 cabinets and I set them at 3 different temperatures, I've confounded temperature with chamber, and so we can't just include chamber as a variable. Yeah, stacking petri dishes is an interesting one as well, Ruby. And again, this is one of those ones where you need to ideally assess light levels and figure out how much light you're losing in your stacks and convince yourself that it's not an issue. To me, I suppose one of the most important things in trying to make these decisions, right? I have had experiments where I submit them and the reviewers say, no, I'm not even thinking about this. You only had 2 glasses and one was warm and one was cold. It's pseudo-replicated. I think most reviewers, what they're actually saying is, can you prove to me that you've thought about this and that you've tried to account for it? So if you match your chambers well, and you measure the starting conditions and you measure the conditions in your chambers repeatedly over time, and you have good records of the conditions that your different chambers have or your different shelves have, and you can look at those and you can logically intuit that the effects that you're seeing make biological sense and that your chamber didn't shut off for 3 weeks in the middle, most reviewers are happy. It's when you don't have any data to substantiate the conditions that you grew your seeds or your plants under that it becomes a problem. Is that making sense?
I guess basically what you're saying is that you're providing evidence that that the chambers are not variable, so that the variation is more likely to be due to the temperature, and that you have evidence that it's unlikely to be due to the chamber. But it doesn't come from that experiment, but comes from other data that you have.
Exactly, that you're collecting at the same time. I think we often forget to collect those data because it feels like, you know, I set my chambers up, I can trust them. But it's always nice to actually have the data, you know, have a few eye buttons on the shelves or do regular light measurements or whatever. But yes, thanks, Terry, that you expressed that well.
Lydia had a question about randomizing within the stack. And I would say that sure, absolutely. I think the idea about this randomization and blocking is our idea is that block whenever you can, because then you're controlling for variation, and after you can't block anymore, then you randomize.
Yep.
So there are strategies for arranging the stacks in a way that's sort of balanced. Randomization on average will make sure that you don't have any pattern, but you can also create design within your stack.
And in the case that the one that you're looking at here, what I said before was that we arranged them haphazardly. And what I mean by that was we randomized them at the outset, but we did not stick to that randomization in a sort of dedicated way. We kept them on their shelf, we kept them light or dark, but we didn't actually randomize. And the distinction I'm making here is you could take all your trays out, do your count, rearrange them and put them back in. You haven't randomized them. You've haphazardly rearranged them. To say that you've randomized them, you actually have to use a randomization process. And so I think it's a useful distinction. Terry, is that a useful distinction to you?
Sure. Jose has said something, and I quite like what he said, so maybe I'll read out that chat. He says, we prefer to control the position in the stack and use that as a variable in the model. Yep. And so that's a very good comment to contrast with the question, should I be just rearranging?
Yep.
Yeah. So yes, the preferred is to is to keep the Petri dish in the same position and then include that in the model in the analysis.
I guess to me, the most important thing is that you think about it. You don't just chuck them in there and assume it'll be okay. You think about it. You think about what the issues could be when it comes to the analysis, and you make a careful choice a priori rather than retrospectively realizing that you didn't account for those sorts of chamber effects.
And I would also say, go talk to a statistician, because they might be interested in your problem, and they'll give a non-biological perspective. And sometimes that's lots of fun to explain your science to somebody who can have an appreciation for it, but doesn't understand, but may have other points of view that could be relevant for your experiment.
So what we're talking about here, we've sort of gotten to this issue of blocking, right? How you arrange your samples spatially. And I just want to make sure that for those who haven't engaged in this conversation that we've just had before, that what we're really talking about is the spatial arrangements of your samples in your experimental design. Now, we've just talked about dishes in a chamber, which is a very small spatial array. What you're looking at here is a rice trial in the Philippines at the International Rice Research Institute. And in that case, it's an irrigation and fertilization experiment where you can't randomize the location of your irrigation treatments, right? One side of that plot, that field, is irrigated, the other is not. Likewise, it's hard to randomize the application of fertilizer because fertilizer moves and there's neighbor effects. So there are designs that you can use along the lines of thinking about where my dish is in the stack or where my stack is in the chamber that can be applied at much larger scales. And these sorts of block designs, or in this case, it was a split plot design, have been designed over years. And there's good sort of papers on the different designs and what their pros and cons are. And as Terry says, a good chat with a statistician can help. To me, the point is, again, think about space. Think about how space affects your experiment before you lie it out. And a brief chat on spatial arrangements in the field. So Megumi has asked the question, are you talking about a random effect? And yes, that's what I'm talking about. So generally, you're aiming to account for these sorts of spatial effects as what we call a random effect in a model. So when you set up your statistical model, you've got your experimental factors. I'm interested in species and I'm interested in temperature, for example. But you also have these other factors, these block factors, or where I am in the stack, etc. And we can treat those as what we call random factors, where we're trying to account for the variance explained by that space or position or time, even though it's not a core to our question, it's something that we want to account for in our design. So we call those random effects. I want to make just a point that when you're doing field experiments, this comes into, we often think of the sort of blocking and spatial arrangement issues as being something that are particular to controlled environments. We were looking at emergence from seeds that we'd put out in the field in that picture that you're looking at there. And what we wanted to know was whether populations from different elevations had different germination when we put them out in the field. We could have done that on one elevation gradient, put all of our seeds on one elevation gradient. But what we did was that we ran a series of transects across different mountains, and we put our seeds out in each of those so that each transect could be treated as a block in the design. And we're looking at that effective elevation on germination across the transect. So we've arranged spatially so that we can account for that variance amongst our transects, rather than putting all of our seeds on one hillside. So when you're doing field experiments, don't forget about those sort of spatial issues. Often we'll do things like we'll set up an experiment and we'll fertilize a plot, or we'll weed a plant, or we'll remove the foliage on a plant, and then we'll look at the effect on emergence from the soil seed bank, or we'll introduce seeds and look at how they germinate. But remember, when you do that, that if you've got a high nutrient and a low nutrient and a control plot and a burnt plot or whatever, you want to arrange those so you don't have all of your high nutrient plots over there in the patch of POA and all of your low nutrient plants over there in the heath. All right, you want to randomize the location of your treatments within your blocks, and you want to randomize the location of your blocks across the landscape so that you're accounting for those spatial variations. And you want to think about bias, because when you're doing field work, and in particular, it's really easy to be like, oh, that's a really nice patch of grass. I want that to be in my experiment. That's not a randomly located patch. That's a really pretty patch. And I think we often have the inclination to say, okay, that's the pretty patch. That's where I'll impose the treatment. That's the ugly patch. That can be my control. You don't want to do that, right? You want to randomly assign your locations to your treatments and think carefully about how you arrange them in space. Because there's so much noise in the field. If you don't do that, you've got no hope. I also wanted to just touch briefly on temperature gradient plates. We heard some really wonderful talks over the past week about new packages to help analyze data in temperature gradient plates. These are a really wonderful tool for analyzing seed germination data, but they're also quite challenging. So in the bidirectional mode, when you set up these plates, and for anyone who's not met one of these, this is a temperature gradient plate. This is the sort of most commonly available brand, although people do sort of make homemade versions. You've got a thermal gradient that runs from left to right, and that can run from front to back as well. And you can moderate the temperatures in time in what's called the bidirectional mode. In the bidirectional mode, where you have day and night temperatures, no two cells are alike. And so this is a replication nightmare. There is no replication for a condition when you're running the temperature gradient plant in the bidirectional mode. And so you have to do all sorts of really creative things to get around that. And I will leave it to some of our other participants who've actually thought in deep depth about that to cover that. In the unidirectional mode, the rows, if you'll call them rows, have a constant temperature, and the columns differ, running in a gradient from say 5 to 30 degrees. And so you end up with this question of, if I'm going to put a bunch of dishes in there, how do I replicate, and where do I put my dishes? We've been using our temperature gradient plate recently to grow plants. This is a challenging thing to do, and I'd be happy to chat with people about how we modify the plate to do this. But in that experiment that you see there, we had 12 lines, 12 different genotypes, and we used what's called the Latin square design, so that every genotype occurred once in every row and every column. And so we were able to account for row and column as random factors in the analysis, and get around the concern that if we put all of a given genotype in one row, we wouldn't have any ability to sort of pick out those special effects relative to the treatment effects of interest. Another one obviously is benches in a glass house, right? If you apply your treatments to your benches in a glass house so that all of your low water plants are on the east side of your glass house, they're also getting a completely different light regime than the plants that are on the west side of your glass house. So think, when you're setting up plants in a glass house, how do I randomize the location of the treatments in space? All right, Now I have pretty much used my time. So I think I will wrap up and say the next 5 minutes. Does that work, Terry?
Yeah, sure. That sounds good.
Okay. What I wanted to do really briefly is fly through an example of how you might kind of put this logic in place. And I'm going to use a study by Anne Cochrane, who did her PhD with me some years ago. She ran the Threatened Species Seed Bank out in WA, and many of you will know Anne. But she had a really interesting question, and I'll tell you a story about the design that we came up with for her and what we learned in the process. So we were interested in how warming and drying would affect germination in 4 species of Western Australian Banksia that occur along a rainfall gradient, and you can see those on the slide. We used plastic funnels, little open top chambers, to impose our warming treatment. Passive warming was really quite effective. We used rainout shelters to impose a precipitation treatment, and we were trying to simulate a future climate in which it was warmer and rainfall had changed. And we redirected the rain that came off of these shelters so that we had control reduced and elevated rainfall. And we started from our hypotheses, right? Our null hypothesis was it won't matter. Warming doesn't matter. Precipitation doesn't matter. The species are all going to be the same. Obviously, that's not what we thought. So we had our specific hypotheses going into this, and we thought through them from the outset so that we could think carefully about whether our design was going to help us to actually test these hypotheses. So we expected the species to differ in how they emerged. We expected that warming or drying would slow and reduce germination. And we thought that perhaps the species from cooler places would respond to those warming and drying treatments differently, or show more detrimental effects than species from warmer and drier places. And that we think species will respond differently to moisture or temperature is what we call an interaction. And for those of you who live and breathe stats, that's obvious. But I think if we all reflect back on when we first came to understanding statistics, getting your head around the concept of what an interaction is and why it matters is one of the most unintuitive, powerful, but unintuitive parts of designing these sorts of models. The other thing that we did from the outset in this experiment is we thought about what our results might look like. Right? We drew lots of pictures of how we thought emergence might differ amongst, say, species, amongst the warming treatments, between the different precipitation treatments. We thought, what are the overall effects and how would that look? And then we asked ourselves, okay, what are the interactions here and what might that look like? So for example, species might respond to warming in different ways. So you've got emergence on the y-axis there. We've got control and warm conditions on the x-axis. And I've drawn one completely hypothetical silly line for each species to show that the different species respond to warming differently. So we wanted a model that was going to allow us to see that sort of difference. Likewise, we wanted the model to help us see whether the species responded to precipitation differently. Unfortunately, our design is what we call a 3-way factorial design, which leaves you with at least a 3-way interaction, which is really, really hard to think about. But we tested it anyway.
Well, Adrienne, sorry, I will say, if you just go back a slide to that one, yeah, that 3-way interaction. Really, it's just taking, so let's say that middle bottom one, the species by precipitation, is taking each one of those segments and splitting it into 2.
Yeah, yep, absolutely.
So that's one way to think of adding that 3-way interaction is just doing another split.
Yeah, so doing 2 panels, right? Rather than attempting to show a 3-way interaction on one panel, which hurts your brain, you could show the species by precipitation interaction under warm conditions in one panel, and the species by precipitation interaction under cool conditions in another panel.
That's
And
right.
that gives you the power to separate those
And-
interactions.
And then we're just looking to see if that pattern that you drew, say in the middle
Mm.
one, is
It's
the same pattern for control as it is warm.
So the experiment that we designed to look at this was fairly insane. We had the 4 species. We included 6 populations of each species to try and get a sense of the within species variation. We had the 3 precipitation regimes. We had 2 temperatures. We put 20 seeds in each chamber, and we replicated this in 4 separate blocks, and then measured mean time to emergence and percent emergence. And when I say we, let's be perfectly honest, Anne did all of this. I gave lots of advice from the other side of the country. I did go visit a couple of times. So Anne's experiment looked like this, right? She had those great big rainout shelters. She had 12 of them, and she arranged them in groups of 3, where the reduced ambient and elevated water were all next to each other, and then you'd walk 25 meters to another part of this, the site, and there'd be another block. So we tried to account for that spatial variation. Under each rainout shelter, we had the chambers and the control, and you can see that all of the warmed plots were in one bed and the unwarmed plots were in another bed, and that was to avoid the warmed plots warming the unwarmed plots, right? If we randomized the location of warmed and unwarmed, we actually would have been warming everything because the little chambers would heat outwards as well. So that was 576 plots and 11,520 seeds to follow the emergence of. Those of you who know Anne know that she's a person of great determination, and this wasn't daunting to her at all. The random terms then that came into this model, right? We had our factors of interest: species, temperature, and precipitation, but we also accounted for block, row, and column, which gave us a really complicated model. But by starting with the big model and then exploring the data and figuring out which of those terms were explaining the patterns that we were seeing, we were able to narrow it down and really focus on the differences of interest. In the end, the experiment actually sort of simplified down to this. The water treatment didn't work because it was one of the wettest years on record in WA, and even our reduced rainfall, which was getting 50% of what landed on the roof, was more than the seeds needed. So water was eventually removed from the design because there were no water effects, but we tested that by exploring lots of different models. The temperature effect was detectable, but the species differed in how they responded to temperature, and that's what you can see here. On average, warming reduced percent emergence, but the slopes of those lines you can see differ. Banksia coccinea down here couldn't have cared less about warming. It didn't have very high emergence to begin with, and emergence wasn't altered by temperature, whereas for the other 3 species, we did see that there was a reduction in emergence, and there also was a slowing of germination under warmed conditions. You can read the paper if you're interested in the biology. It's a cool paper. My point in running through that very quickly, so I'm sorry for having done that so fast, was to get you thinking about the model that you use and how it can help you account for the spatial issues, the replication issues, and help you make decisions about where to put things in space so that your replicates, your seeds, are doing the most work they possibly can for you. And I also really want to stress that working out what our predictions were, what we thought the data might look like from the outset, just with silly pictures drawn on bits of paper, helped us frame the design and think about what the model would ultimately be.
Adrienne, there's a question from Ruby in the chat. Did you use model selection?
So Ruby, this experiment predated the common application of model selection. We worked in GenStat, not R. And we didn't do a formal model selection. We started with the full model, and then we dropped terms that weren't significant. AICs weren't in common use at the time. And the other thing that we played around with quite a bit was that population effect. We did have a priori interest in the differences amongst the populations, but we explored a bit whether we wanted to treat them as sort of a factor in themselves, or whether we wanted to use them to increase our replication and use them as another random factor. So we did model selection in that we explored lots of different models and thought carefully about which ones we wanted. And in the paper, we explain exactly what that process was, but we didn't do sort of an AIC approach to selecting a model.
I noticed also that Jeff Wood, one of my colleagues, is on that paper.
Yeah. Yes. We had lots and lots and lots of long chats with Jeff and Anne. It was a really complicated experiment. I don't actually recommend doing something that complicated. The temperature gradient plates, yeah, Meg, I'd be happy to chat with you about that. We just use seedling raising mix, and we grew the plants in little baked bean tins, baked bean, corn, chickpeas, because the tin carried the temperature effect most effectively up through the soil. And we tried lots and lots of different sorts of pots, and in the end, baked beans was the way to go. All right, thank you all so very much. My ending point, and where I pass to you, Terry, is just to really encourage people, once you've done your experiments, once you've collected your data, get to know it well, become a friend of it before you chuck it into a model and send it out to work on its own. Getting to know your data and having a sense of whether you've entered it correctly, whether there are mistakes in it, whether there are really weird critters that are doing something completely different, which, as you all know in seed science, you can sometimes get an accession that behaves really differently from all the other accessions of seeds. And when you really look at it, it's because it's a completely different species. So you really need to get to know your data and think about what it's doing before you set it free on the stats. And I know, Terry, this is something that you care deeply about. So perhaps it's a good time to pass over.
Thanks, Adrienne. That was a really great segue into my section, which is coming up. So I think that, I don't know if you can see me. Maybe if I turn on my video, you could see me. So I think that we'll take a bit of a break, but for those people who are new and feel a little bit uncomfortable with R, I'm going to show you how to set up a project and to make it easy to import your data. So why don't we for those of you who have already set up a project, then you can take 15, yeah, come back at 10:15. And those of you who haven't, will get you all set up. Great. Okay, so I'll go ahead and start by sharing my screen, and please just talk. Yes. So there's a lot of thank yous and applause on the chat, and I also really enjoyed your talks. Thanks very much, Adrienne.
Thanks, Terry.
I'm going to go ahead and share my screen. Let's see if that works. And I might take that off. Yeah. So I have an RStudio up. I've just opened RStudio. And right now I have three windows, three windows open. The first thing I want to do is I want to run my R code within a directory. Why do I want to do that? It's going to make my life much easier. I'll be able to access my data easily. I'll also be able, whenever I create outputs, creating new data sets, creating plots, they will automatically go into that directory. So you'll have within your project all of your input data, you'll have your R code, you'll have your output, everything will be together. I think it was Lydia who mentioned that what we really want to be able to do is to create reproducible and transparent code, not just for others, but for ourselves. So when we come back to the project 6 months later, we'll just open up inside our project, we'll be able to immediately access the R code. I talked to a lot of researchers and really appalled at some of the practices about the lack of respect that people have for their data. So they may just keep their data someplace on the desktop and maybe not remember where it was, 6 months later. So what we're going to do is set up an R project. So those of you who haven't done this before, here is my R Studio, and I hope everybody has their R Studio open. I hope that everybody has downloaded their data set into a directory and hopefully also their R code. But I'm going to start with just having the data. What I have done is I've put the data set into a directory. Okay, I'm going to call that directory is going to be where my R project tab sits. How do I do that? I come up here to the upper right corner. You can see here, hopefully, it says project none. So I'm going to set up a project and I'm going to put that bookmark essentially into the directory where I have my data. So I click on it and I get this tab. And the first thing that says new project, okay. Create a project. I'll click on this and a window will open up. I hope you can see the window. It asks two main questions, new directory or existing directory. Now, because I've already put my data into a directory, I'm just going to go find that directory. So I'm going to click on existing directory. And Adrienne, can you see all these windows that come up?
Yes, but I can't unmute. Yes,
Okay.
it's all looking good, Terry.
All right, so it's all just a matter of going to find it. Now I've put my my directory where I kept my data inside Dropbox, and I put it in workshop 1. So I don't know if you can see my workshop 1, but inside I've just got the data set. On the bottom of that window, there's a button that says open. So I'll click open. This will tell me, this now tells the R what the directory is, and I will click on the button that says create project. Okay. I hope I didn't do that too quickly. So now when I look in the upper right, I see that it has a name, workshop 1, which is inside my C directory. And I have my R project here, and I have my Excel file here. Yeah. So I would like to really make sure everybody has this set up. Every participant is prepared to do the R project, because if you can't enter the data, then really you won't be able to do anything at all. Okay, so it'd be good to go. We're officially starting at 10:15, but it would be good to get an idea from participants if you could just hit your reaction button as to whether you're ready to go and you're up at this stage. Let's see if I can see participants. Okay, I see a few thumbs up. Great, so a thumb up is a great thing. I'd like to see a few more of those. And if you have any questions, just speak up and we'll try to solve your problem. The setup is everything.
Terry, what stage are we supposed to be up to? Sorry, I just was away from the computer.
Oh, right. Okay. So where you should be is, if you can see my screen in my RStudio in my 3 windows.
Yeah.
If I'm looking in the upper right corner where it's where there's a little R insignia.
Yeah.
I should have set up a project.
Yeah.
So you can see down in the in my files tab down below, I've set up a project called Workshop 1.
Yeah.
And I have my data.
Yeah.
Okay, so that's where everybody should have this. This way I'll be able to access my data very easily. And this extends for the whole day, because Sergey, I should I should say that this is Sergey's workshop. Okay. Lydia attended Sergey's workshop and she was really inspired. She said, let's replicate that here. I use my own R dialect, but basically all of the content comes from Sergey. So I need to give him full credit for this workshop. And I'm really just doing this because it's the middle of the night in Germany. So I'm using Sergey's material basically, but just so giving him a chance to get a little bit of sleep before he comes in the afternoon. Okay, so are there any other people? I'd like to see a few more thumbs up. I don't know why my thumbs up just kind of disappear. Is there, I see another thumbs up from Ruby. Anybody having difficulty? If you're having some difficulty, you need a little more time, maybe you could either just tell me or put in I think there's a go slower button, right?
Terry, it could also be if a couple of people already had projects, they might have gone off on their break. So that might be stopping them voting.
Okay. So what time did I say to come back?
1:15.
1:15, okay.
Terry, I saw in the email that it said we had to have version 4.11 or something. Does that matter? Is there a difference at all?
Well, you can see I have 4.0.2, so I forgot to do that. But at that level, I think it's people who have version 2.7.2 are the ones we have to worry about. Yeah. So the important packages. There are some. I think what happens is that people write code and they put it up in the repository, and some of that code will be compatible with the latest version of R, but may not be back compatible to much earlier versions.
Okay.
Okay.
Yep.
So I'm glad you mentioned that, because that was something I meant to do.
I can confirm that I had a very old R and the code did not run properly, so I did have to upgrade. But if you've managed to run stuff and not get errors, then hopefully you're okay.
Right. Yeah.
So they don't make major changes with each version they make. They're usually pretty similar.
Well, yeah, they're incremental changes. So they tend to be back compatible up to a point. Yeah. And I think so that's relevant if you write code, and I don't know, I'm the statistician and I'm not a coder per se, but I think when you put your code up and you put your data set up, it's important also to say what version of R it ran on. And it is possible to be able to run things on old versions, but that this version is a small issue. I think that we're reaching a point where I hope things are going to be pretty back compatible.
So we might give you a quick break there, Terry. If you want a couple of minutes, we'll stop the recording, please, Aranet, again. I think I can see it going already. So yeah, we're good to go, Terry. Thank you.
Great. Okay. So line 2 is not important if you've already done this. You only have to install your packages once. Yeah. But I'm assuming this was part of the instructions. So I'm assuming that you've done this step, but in case you haven't, make sure that you run this code to install your packages. Great. I want to give you also a little bit of discussion about how we write code. And we do, and in order for us to be transparent and reproducible. Yeah. So when I write a comment, you can see when I write the hash that it goes green, which means I can write comments. A slightly nicer way to write comments is the hash with a single quote. And here I might describe the background of my experiment. When I hit return, you'll notice that I get my hash and single quote back, which makes it easier for me to actually write in paragraphs, okay? So my advice is to use my comments liberally, yeah, so that you'll be able to follow your own code and other people will be able to see what you do. The other bit of etiquette that I encourage, you see I'm going to have to hit delete to stop that comment. The other bit that I encourage is to write my code in a modular way. In other words, I want to be very clear about the steps that I'm going through. What are the steps that I'm going to be going through? And maybe I just specify them in this. So my first step is going to be to load my libraries so that it's very clear which packages that I'm going to be using. And I'm going to load these 4 packages, okay? My next step is to import my data. And each of these is going to be then a separate paragraph. My next step is something that I call data wrangling. I guess you might call it data manipulation, but manipulation just has negative connotations. So I may want to restructure my data. I may want to change some of my data types. And R has a lot of very powerful tools to rearrange your data in a way that turns it into what we call tidy data. That's easy to look at and easy to model. Okay. And step 4, and that's as far as we're going to go in this section, is what I call exploratory analysis. And that really is data visualization. It's so important to be able to look at my data, to try to understand how my data are distributed, to try to understand the variation, to pick out outliers, pick out weird points that may influence my model. So we won't be driving blind when we start modeling the data. Modeling is the subject of Sergey's afternoon session. Okay, so I'm going to take us through these 4 steps. So what I'll do actually is, well, maybe I just keep this up. And let's begin with step 1. So, and I may in fact write this, load my libraries. Now, typically when I set up an analysis, I don't always know exactly where I'm going. But the package that I use all the time, which I ask you to use, is tidyverse. Okay, so tidyverse I want to think of as a dialect of R. That it's actually a bunch of packages that are put together that will give you a lot of power in a relatively small vocabulary to be able to do your data wrangling and do your data visualization. Okay, so tidyverse is stock standard, and we'll be using the language of the dialect of tidyverse in this in this session. Okay, and the next library I definitely know I need is package that I definitely know I need is I need a package that will allow me to read Excel files. and that's called read Excel. Okay. Now, to run to run my code, the only thing I need to do is make sure that my cursor is anywhere on that line. Okay. Then I can come up here and press run, or I can press Control Enter. Or if you're on a Mac, it's Command Return. Okay. But because I can't really show that on the screen, I'm just going to hit this run button here. Okay. So this first one will run the tidyverse, and it sends me some messages. Notice that it's going to be attaching in particular a package called ggplot as well, which is what I'm ggplot2, which is what I'm going to be using for data visualization. So I don't need to load that as well. And I will also run library Excel. And now you'll notice that each time it just in my console. So up here is where I write my code. Down here is my console, is where my code gets run. So this console is really the engine of, and it's asking for prompts. I could write directly into the console, but then I wouldn't have it, right? So this is the code that I'm going to have, that I'm going to be able to save, that I'm going to be able to run again. Okay. So I could, for example, go back and run this command again, and it will be happy. And I can run this one again, and it will be happy. It will return a prompt which says, give me more. Okay. So this is what all I'm going to have for this first module. In my second module, what did I say? Step 2, import the data. So I'm going to import my data. Now, this is where it's been very useful I can see my data here in my Files tab. Yeah. So that means that this code, and I think what we'll do before we do anything else is we'll save this code and we'll give it a name. Okay. I think I will call it what Sergey called it, which is the warmup.r. Okay, let's save this file. And you'll see that now this R script appears in this directory. Okay, so as I'm writing in R, it will always be looking for things in this directory. Okay. So I want to introduce you again. I can't show you what I'm doing on the screen. So there's this idea of tab complete. Tab complete is a wonderful tool that coders use all the time. And basically it means that if you press tab, you'll get some choices. Okay. So I'm going to use this tab complete to be able to find my data. Now, in this case, it's not an issue because my data has a very small name called data, but I'll show you how this gets used. To import the data, I have a command called read Excel, okay, or a function called read Excel. So that will import the data, but I want to give the data a name. So R is an object-oriented language. So every object, in particular a data set, has to have a name, and then we can refer to that name. Okay, so what I will call my data set, and I'd like to give it short names. The name of my data set that will be in R, I'm going to call germ1. This command, I could write it equals, but so we either write equals or we write assigns, and that is the shift less than followed by the, I don't know what you call it, slash, horizontal line. Yeah. So it looks like an arrow. So hopefully everybody can get that. Yeah. Sometimes I put spaces, but the spacing actually doesn't matter. Okay, just put spaces to make it easier to read. Okay, so this file, I'm going to import my data. So I'm going to use this function read .xlsx. You can see as I'm typing, it offers some suggestions. Okay, I haven't used the tab complete yet, but now I will. I'll put in double quotes, although you can use single quotes. This is just my convention. Now I'm going to hit the tab button. And what it will, I hope you can see that, but it comes up with all of the possible, all of the files in the directory. Yeah, so I can just choose data Excel. So if your data set had a long descriptive name, then you wouldn't have to write that had spaces in it. You wouldn't have to write that all out. Okay. Now this data set that Sergey prepared has a number of sheets. If I just write this code as it is, it will by default download the first sheet. But it's actually good practice to specify the sheet. Sometimes the sheet has a name. In our case, I don't think it does, but we just will take the first sheet. Okay, so we'll write sheet equals 1. I don't need the word sheet equals. It will understand if I just write comma 1. So we'll read in the first sheet and we'll give it a name. Now, everybody should get to this point, and it would be good to have some thumbs up if you have created an object called germ1, and that will appear in your environment tab, hopefully in the upper right corner of your, yeah. So I'll look for your thumbs up. And Lydia, maybe you can just let me know when it's okay to go on.
We've got a few thumbs up, so I think it sounds like people are good to go.
Yeah. Is there anybody who's having trouble who
Haven't seen anything in the chat. Oh, we just had a question pop up actually from Daniel asking, is there a difference between working with Excel versus CSV files?
Well, from our point of view, no, but there is a command. If I want to import a CSV file, I'm going to use the function read_CSV. Now, a CSV file, we don't need to specify the sheet. So I think that if you have run an experiment and you have, but you have several sheets, I would tend to, you could either have several CSV files, or you could have all your data on a single, in a single workbook with multiple sheets. I think it really depends on how you think it's the best way to organize your data. But from R's point of view, so the old R, before we had this library called Read Excel, would only read CSV files. Nowadays, R can read Excel sheets, CSV files, SPSS files, SAS files, Stata files. I can't think of others, but it can read in a range of data file types. Okay, so the first thing I want to do is explore these data. Okay, so what are good ways to explore the data? One way I want to make sure I have the data that I think I have, I can write a function called structure, which its name is str, stands for structure, germ1. Okay, I'll run that. Press the run button. And this tells me the structure of my data. Now, this data set is very clean. Not everybody has nice, beautiful, clean data, but my original data has in the first row the names of my variables, and I have one row per dish. Okay. I notice that I have 300 rows and I have 11 columns. Each of these columns has a name, and each of these columns is also an object in itself. Okay, so I could, and for example, the object species, which his name is not species, his name is germ1 $ species. So it's kind of like you have to give the family name as well. But if I write, for example, germ1 $ species, then, whoops, didn't run it. Then I get that the 300 values, starting from 1 down to 300 of the values of that, okay? And that is actually also an object in itself. It's what we call a vector, okay? But let's go back to my structure statement. And there are some important things to gain from that, okay? So it gives me the names of each column. I have, and we talk a little bit about this experiment. I have some number of species. I have run this germination experiment across a range of temperatures. Yeah. The seeds variables tells me how many viable seeds were in each of my dishes. And the rep is just the name of the replicate for a fixed species and temperature. So you can start to see it looks like 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So it looks like I've got replicates. I've got 5 dishes for each species and temperature. Okay, but those are questions we need to do a little bit more exploration. Then I have these columns called D1, D0, D3 up to D31. So this just refers to the number of germinated seeds that were observed at each of those days. Okay, so this is actually, I think the data were arranged this way is because this is a very common way to arrange germination studies, and it will use this data set when we do use a germination specific package, Germinar. Okay, and this is a very good way to arrange your data, to organize your data, because often this is how we might write this down in a sheet. Okay. Now I may want to look at my data. Let's go over here to the upper right hand side. And I can press in 2 different places. If I press in this green button here, or the blue button, then I'm going to get what I got for structure. And if I touch on the data set, then I can actually see my data. Yeah. So one thing that people often do when they first start using R is they think, oh, I have to go find I have to open it in Excel, then I have to open it in R. Actually, this allows you to see your data as an Excel spreadsheet. You don't have to use Excel, just click on this. Now, you'll notice that in the console, it wrote the words view. So that would be another way that you could view your data, is just type in view germ1. Okay. I can keep this and I can just go back to my original tab. I talked about the names of the variables. This next column here has these words CHR and num. They just stand for the data type. And those of you who've used SPSS will be familiar with this idea of data types. We have basically 3 different data types. We have character fields, which are just text fields. We have numerical fields, which are numbers. And we have what we call factors. Okay, so a factor is our discrete values. They're like character fields, but they have more structure to them. So for example, I may have 5 different, 10 different species. So the species would then have, I would have 10 different levels, and each level would have a particular name. So it's like having a little box. So it's discrete data rather than just an unstructured character field. Okay, and so we can take character fields and we can turn them into factors. Sometimes we have numerical fields, we want to turn them into factors. Sometimes we want to turn a factor into a numerical field, and we have functions that can shift, that can change those data types as we need to. Okay, and we'll be playing with those as well. Another function I can look at to look at my data is a function called summary. So let's write summary germ1 and see what we get. So summary germ1 will give me, and I might just push this up to get a full idea. It will summarize each variable. Now, when it has a character field, it doesn't really give you much information because it's just a text field, so there is no structure. For the numeric fields, for example, seeds, it will give us some summary statistics because it's a numerical variable. Yeah, the minimum, the first quartile, the median, the mean, the third quartile, and the maximum. Okay, and you can see for something like rep, you kind of think you'd want to be a factor, although because we won't be using it, we won't be changing it. And we can also see what's happening at day 0. There's no germination. We can start to get an idea of how each day, how the data are distributed. Okay, again, a first look. This is a place where we can start to look for missing values. We could start to look for unusual values. Okay, so we want to look at this. One thing we haven't done though, and something that you can't tell from this so far, is how many species we have and how many temperatures and how many temperatures per species, etc. Okay, so that would be another way to get an overall idea of what's happening with these data. So now we're in the section called data wrangling. And I should put my little single quote here. Okay. And in this section, we're going to make use of tidyverse and some of the power that it has. So we'll be using functions from tidyverse. Now, one of the beautiful parts of the tidyverse language is something called the pipe. And this really comes from the computer science world, computer programming, because coding people got involved in R, so they weren't just statisticians, and they made our language really much more powerful. So the pipe is this idea that when I write code, I'm going to take what I have, some existing object, and then I'm going to apply some function to it, and then I may apply another function to it. So it's like doing a series of steps one by one, and each time I take what I have got and I apply something else. And the thing that connects that is the thing called the pipe. And the syntax is a shift 5, shift greater than, shift 5. Okay. So whenever you see this pipe function, you can insert the words and then. Okay. And I think Sergey has given you some has given you some links to get some more information about using the pipe. Okay. So what do I want to do? Well, what I'd like to do is I would like to summarize my data. I'd like to know how many dishes do I have for each species and each temperature. Okay, so that's the question. How many dishes per species? Temperature. Okay. So basically, I want to take my 300 observations and I want to summarize them and just count how many rows I have for each species and each temperature. Okay, is that clear? So I'm going to start with my data set. that was called germ1. Okay, I'm going to pipe that. So, and then what am I going to do? And I have a function called group by. Group by is a very powerful function. Basically, I want to summarize by species and temperature. So my group by is going to group by species temperature. And then, so pipe, I want to summarize. And what do I want? I just want to get the number of rows for each of those combinations. So this combination, group by and summarize, can be used to great effect. Right? I can summarize for each species and temperature, the mean number of seeds, the mean number of germination on day 3, et cetera, et cetera. This time, all I want to do is count the number of rows. And so I'm going to call that variable count. And what count will be is the number of rows. And the way we write the number of rows is just n and open close brackets. That's it. Let's have a look. I'm going to run this. And you can see I've created a new data set. Now this data set has only 60 rows and 3 variables. The variables are species and temp, because that's what I grouped by. And then this new variable that I've created called count. So I can see now that there are, well, it looks like there are 6 temperatures. And well, I could deduce actually that there are 10 species, because each species probably has, I could see this if I wanted, I could give this object a name and then look at it just by clicking on it. So I would create a new object over here. But I think maybe an easier way to look at it would be really nice if I could put species as rows and put the temperatures across the top. Yeah. Okay, if I could just put temperatures across the top and I could put the counts inside. That would be a nicer way to look at these data. So we actually have a beautiful function. I'm going to restructure my data. I'm going to make this data set wider, okay? So that I'll have a temperature. Each column will be a different temperature. Each row will be a different species. And what will be entered are the counts. So let me just show you how to do this. And this is a function, again, that I encourage you to read up more about. It's a function called pivot wider. This is a very clear name that we want to take a long data set, which is 60 rows, and make it into a wide data set. Okay. So pivot wider requires 2 things. It wants to know what I want to make wider. And that argument is called names from. So it gives me some hints, names from. And I think I can just put without quotes. Yes, names from the variable called temp. Yeah. And it wants to know what I want to populate that table with. So those are called, that's called values from. And hopefully, if you're getting it, you'll know that values from comes from count. Okay, so I'm going to populate my data set with the count. Let's have a look at what this creates. I'm going to hit run. And now I've got something that's much more meaningful for me, and I can see immediately that there are 10 species and that I have 6 temperatures and that I have a very balanced design. I've got 5 dishes for each species temperature combination. Notice this is also a data set that I've created, and I could give it a name and then it would appear in my global environment. So is everybody able to get this table? Oh, okay. I didn't know about the shortcut for pipe. So Control-Shift-M. Is that is that for a Mac or is that for a PC? It works on a Mac. Okay, great. Okay, thank you very much.
We had a few thumbs up, Terry, so I think people have followed along to this point.
Great. Okay. So now we feel we have a kind of a sense of these data. Let's look at these data. So now I'd like to go to my one of my favorite sections, which is called what I'm going to call exploratory analysis and data visualization. Okay, so make sure that I'm on the same, right. So what would I like to do? Well, if I really wanted to visualize these data, let me go back and have a look at just pull up my data again, pull this down a little bit. So you could see that each row is a dish and that we've measured the days across. So what I kind of imagine is how I might want to visualize these data is I might want to put day on the x-axis, right? And then I could follow a dish over time. So time basically is going to be on the x-axis. And what am I going to measure? Well, I'll measure the number of germinating seeds. But the data are not set up at the moment to be able to visualize this because I have multiple columns for day. What do I need to do? I need to create one column for day. So so how many days? I have 7 different times. Yeah. So I'm going to take these 7 columns and I'm going to turn them into 2 columns. One column will be the day column, and the other column will contain the number of germinating seeds. Now, remember we talked about this guy called Pivot Wider, which takes a long data set and makes it wide. This time I have a wide data set and I want to make it long. Okay. So I'll be taking these 7 columns and turning them into 2 columns, a time column and a germination column. So we'll go now from 300 observations to 300. So why doesn't my germ 1 appear as a tab on the upper right? You don't have anything in your environment, Adrienne. I don't know if I can answer your question. Maybe you don't have the environment tab open.
No, I do. I was asking the hive mind. You should keep going, Terry.
Okay. Okay, but everyone is with me. I'm going to take this data set and I'm going to rearrange it. So I have 1 column for time and 1 column for numbers. So right now my data set is 300 observations because I had 300 dishes. But now I'm going to turn my data set into something long. So it will be 300 times 7, yeah, or 2,100 observations. Great. Okay, let's do that. Now, I just realized this, right, that I need to do this. But I think of this manipulation as a data wrangling step. So I'm going to go back up and put it in my data wrangling module. Okay, and I'm going to create this new data set. So I'm going to give it a new name. Now I'm not very creative, but I tend to choose, but I want to choose a name that's going to describe what I've done. So I'm going to call that germ1_long. And I'll use my arrow. So less than and bar. And what's germ long going to be? Well, we start with germ 1. Okay. And then we're going to rearrange it. So you might not be surprised that this function is called pivot longer. Okay, we have pivot wider and pivot longer. The first thing I need to say is which of the columns I want to rearrange. And I want to rearrange those day columns, right? So I could specify those. So I have something called cols, which is the columns. Now I could write the names of my columns, but we have sometimes we can use some shortcuts. So we're going to use it here. So whenever my column starts with the letter big D, because luckily that's how my data have been arranged, those are going to be the columns that get reorganized. Okay. And I would say that this writing it this way is more reproducible than writing out all of the columns, or we could write columns 5 through 11, something like this. But it's harder then for people to then say, oh, which were columns 5 through 11? So if I write this, and people are familiar with the data, they know that I'm taking all of the day columns. Now, when I write a pivot longer, or when I write one thing I often do, rather than writing very vertically and all the way off to the right, after a comma, I can hit a return. It makes it, again, easier to read, and also for me, easier to write. Okay. Now, if you remember in pivot wider, we had a names from and a values from. Well, you may not be surprised to know that we have something very similar, namely a names to. And this is what names to is now what I want to call my time column. So I'm going to call that day. I'm going to put a comma. I could keep writing, or I could hit return. Then I want, rather than values from, values to. So I want to say the name of the column. I'm creating a new column, and it needs a name. And this is the name for the column that's going to contain the germination values, the germination numbers. Okay. So I will call that germination number, and I'll write it this way, germination number. Okay. Great. Well, let's have a look and see what we've created. I'm going to run this code. And those of you who have successfully run this code will see that you've created a new data set called germ1 long that has 2,100 observations. That's the 300 times 7, and fewer variables, 6, because it's taken 7 and turned them into 2. Yes, let's have a look at it. So I can look at it by clicking on it. And you can see now that I've created a column called day. And now I've taken, which has taken the names of the columns and it's just put them down, and germination number. Yeah. I could also look at the structure of this data that I created. So I'll go str. And I would recommend that whenever you do a data manipulation, is that you go and check to see what it's done to make sure it hasn't done anything crazy. Yeah. So I can see that I've got a column for species and temperature, and for seeds and rep. And now I have the, instead of the day 0, I have a column called day. Notice it's a character field. This might not be great. I might want to make this into a numerical field. This could be an issue because it's hard to turn. If I just turn it into a numeric, I'm going to, it will see that there's text and it will just turn it into NA. Yeah, so I'm going to have to remove that D and then turn it into a numeric variable. And again, I have this column called germination number. Okay. So the beautiful thing again about pipes is that I can add things on. Yeah. And I can always just repeat what I've done. So when I code, one of the things I like to do is do something that I know works. And if that works, I might make it a little bit more complicated. Okay. So what I'm going to do is now what I'd like to do is just remove the D from that from the data. Yeah, remove all those D's. So it just says 0, 3, 7, etc. Okay. So fortunately, within pivot longer. Okay. So all of this is contained within pivot longer. So I'm going to, after the germ number with the quote, I'm going to put another comma. So I'm still inside my pivot longer function and hit return. So notice this parentheses is connected to that parentheses, right? So inside my pivot longer function, I have another thing called names prefix. What's names prefix? It's telling the computer that actually I want you to trim off something. In particular, I want you to trim off the D. Yeah. Let's have a look and see what this does. So I'm going to look at my germ long. Now I could just, if I have it here waiting for me, I can just look at it here and you can see it's just trimmed off the letter D. However, when I look at the structure, and I don't have to write it because I've already written it. When I look at the structure, even though it's trimmed off the D, it's still a character field. So how do I make it into a numeric field? I know if I make it into a numeric field, it will give me the right numbers, okay? Because these are just now numbers. So it will just turn them into numbers. How do I do that? I'm going to pipe what I have now, and I'm going to use a function called mutate. Okay, it's part of our vocabulary. So far, we've learned group by, summarize, and now we have mutate. What does mutate do? It either creates a new column or it overwrites the existing column. In this case, I want to overwrite an existing column, the column called day. I could have given it a new name and then it would create a new column. The function I'm going to use is a function called ASNumeric. And ASNumeric turns a data type from a character field into a number field. Okay. So this is the code that I'm going to be using. Let's run this. Okay, let's run the structure statement. And now I can see that day has become a number. Okay. If I look at my command, there isn't a whole lot written there, but it has, in a very powerful way, rearranged, restructured my data. So now I can look at it. Are there any questions about this? Okay to go on? I can see you, Gemma, so great. Okay, so let's go on. And I want to introduce you to ggplot, which is something that, you know, I have to say ggplot, I have to say when I first started using it a few years ago, it felt a bit awkward. But I think it's a lot like speaking German, you know, like if you're learning German and you're not a German speaker, then you don't, you know, putting the verb at the end of the sentence doesn't quite make sense. But once you learn how to speak, and you're learning how to speak ggplot and you're learning what they call the grammar of graphics, then it starts to become more natural. We'll go through the first one a little bit slowly and I'll take you through the syntax. Okay, so basically we're going to use this function called ggplot. The first argument in ggplot, and it gives you a few hints, is you want to specify which data set you're going to be using. So you always have to use ggplot will only graph data sets. My data set that I'm going to be using is my germ1 long. Okay, germ1 long. Now, if you want to make it clearer, we could write data equals, but it's not necessary. It's always expecting that the data set will be the first argument. The second argument is describing your layout, and that's contained inside of a function called an aesthetic. And so we type AES for aesthetic and the open close brackets. So inside that function aesthetics, I'm going to specify what's going to be on the x-axis and what's going to be on the y-axis. Now, because I've set up these data, and I might just put the structure up here so we can see that very clearly. I have day will be on my x-axis and germination number on my y-axis. Yeah, so always the response is going to be the thing on the y-axis. So let's just write that. Now I write x equals, but you don't have to write the x equals. It expects the first argument to be the x. So x equals day, y equals germ number. And because I'm within ggplot, it's looking for the names of columns. I don't need to put quotes in there. That was just germ number. Okay, so if I were to run this, I would get nothing because I haven't specified what kind of graphical object I want to use. So the next thing I'll do, I put a plus, and again, I tend to always, after a plus sign, just like a pipe, I tend to hit return, and so that my ggplot tends to look sort of more like poetry, like a stanza of poetry. Okay, so now I need to specify the geometric object I want. Suppose I want points, then I write geom underscore point. Okay, and now I will get something. Okay, so this code, I don't know why it's sending me an error. It shouldn't be, there shouldn't be an error. I should be able to run this. And I get something that maybe I don't like so well. It's a bit hard. So what's the problem with this? Well, I've got time on the x-axis. I've got germination number on the y-axis, but there's probably a lot of repeats. Yeah, I can't really see patterns. And also, I'm really interested in what's happening between species. I haven't separated out species. I haven't separated out temperatures. So basically, I get something that's not particularly meaningful. How could I get something that's meaningful? Well, maybe I want one of these plots for every species and every temperature. Yeah. So I can do that, and maybe I'll get something more meaningful. So I have another command within ggplot to create multiple plots very easily. I'll hit plus, and there's something called facet grid. What have I done? So I'm going to, now I'm not, yeah, it depends on which order you want it to be, but I'm going to put species. I'm going to use that tilde function, which is in the upper left of your keyboard, and temp. Yeah, so that's it. This will create a grid, and species may be on, well, I'm not sure what it will be. So it's put species as the rows, and it's put temperatures as the columns. Yeah. So now I can see what's happening for each one. Okay. Now I could reverse that and put species as columns, and you could see I'm just going to reverse the role of species and temperature. Now this is okay, but it's a bit hard to read. You could hit the zoom button and you would be able to see it more clearly. Yeah. But one thing that we haven't done is we have connected the divs. In other words, we have longitudinal data. It would be really good if we could maybe draw a line so that we could see what's happening for each dish longitudinally. How do I do that? Well, I have a function called geomLine. I can create lines. Yeah. The only thing I need to do is specify what are the things that go together. And that, if you remember, for each species and temperature, I had something called rep. Yeah. So if I, and I'm going to have to put this inside an aesthetic. So I'm not going to try to explain that, but if I put it inside an aesthetic, it will look in the data sheet for a variable called rep. And I'm going to group by rep. Okay. So what will this do? It will draw, and make sure I put a plus sign here. Why have I ordered it this way? The ordering will, the way the ggplot does things is it does things in layers. Yeah. So sometimes the ordering will matter. For example, if I put a box plot and then I put geom point over it, if I put the box plot first, then the data points will be superimposed over it. And if I do it the other way around, the data points will be behind the boxes. So it works as a layering. In this particular case, I don't think it matters, but it's something that you could play with, the order in which you put things. But basically think of this as we're layering. Okay, let's have a look and see what we get. Okay, so this starts now. We can start to begin to see the patterns of our data. Yeah, for each individual, and each one of these little facets, so each one of these little plots contains 5 sets of lines, and you'll be able to see how well it replicates. So again, we're starting to understand our data. We can see that some species really don't germinate very much. Others germinate a lot, but some germinate late, some germinate early. We can see that we have a fair amount of good replication between dishes. But let's suppose what we really are interested in are temperature effects. In other words, what I'd really like to be able to see is what's happening, for example, in this row, and be able to, one thing it's hard to do is really see those trends across temperatures within a species. How could I look at this? Again, we're still exploring our data. So what do I want to do? Well, I would like to put all of these guys and collapse them onto one graph. But of course, once I collapse them onto one graph, then it'll be hard to tell which is the right temperature. So I'm going to color it by the temperature. Okay, let's look and do that.
Terry, before we create that plot, I might just read out one of the questions in case other people have come across this, but there's a general thing similar to Adrienne's earlier where sometimes, for example, figures don't pop up where you expect them, or data's not popping up where you expect it. So in this case, somebody's got the plot tab not showing the figures. Is there any general advice people would like to offer? I know Caroline's responded with a bit of code that you could try. I'm not sure if that's solved the problem yet, but I just thought it might be worth discussing briefly, if that's okay.
Yes, well, I'm hoping somebody else has a solution, because I don't. This is where I'm a little bit lost. Yeah. So it could. I mean, sometimes when I get misbehaving graphs, sometimes I restart my R.
That's a good tip. All right. Well, we can continue in the chats and breakout rooms and help solve that one. But just thought I'd mention in case other people had similar problems.
Okay.
Seems like we're good to go.
Okay. So I want to look at temperature effects. within species. Now, one thing that's quite nice is that I can do a lot of cutting and pasting, so that I don't have to necessarily write my code again. Okay. So, because I'm still working on the same data set, I still want day to be on my x-axis. I still want y to be on my, I want germination number to be on my y-axis, so that hasn't changed yet. Instead of facet grid, though, I'm going to use facet wrap. And because I want to, so facet wrapped now, I want to have a different graph for each species. Whoops, so I write, sorry, that tilde again with species. Now, oh yes, and then I said I wanted to color things so that I get a different color for each temperature. So hopefully I can start to see the effect of changing those temperatures. So up at the very top on this line here under my aesthetic, I have another possibility, and that is to specify the color. Okay. Now, if you look what I have so far, I have day as a factor, temperature as a factor, and species. So in effect, I can look at 3 factors at one time and start to really understand my data. Now, I just have one problem here, and that is that variable called rep, if you remember, is just 1, 2, 3, 4, 5. So that's okay if I'm working in this facet grid where I have a graph for every temperature and species. It's not going to be okay when I start to combine my temperatures. Yeah, so I really want to have a group for every rep and temperature. Yeah, so the way I write that is I'm going to create a new object called rep temp. I may have to make sure that I have enough brackets. Yeah, and the computer guides you as to which bracket you're up to make sure that your brackets are matching. Otherwise, the code won't run. Okay, so this says I'm going to create a group for each rep temperature, and that then will refer to each dish. Okay, it would be nice if I had a column that says dish number, and then my group would equal dish number. Okay, so let's run this code and see what we get. Okay, so if that ran well for everybody, you can see that I have a different plot for each species. Temperature now is going to be we get a legend for temperature, and we get 5 lines for each temperature. Yeah, I could get rid of point if I wanted. If I wanted to know, well, I wonder if point's getting in the way. I can just simply, because I've written my code this way, if I comment out geom point, then everything will still run. Okay. And the only thing that will happen is, I don't know, I've still got points somehow, is those points should disappear. I don't know why that didn't work. Try that one more time. Yes, okay. So maybe it gives you something slightly cleaner because maybe we don't need the points, yeah? But we start to now look at this particular, now this really allows us to focus on the effect of temperature and the effect of temperature and how it affects different species differently, perhaps, yeah? So we can see that there is definitely a strong temperature gradient for this species, but maybe temperature doesn't make a big difference for this species, yeah, or this species. So sometimes it has the effect of delaying germination. Other times it may have the effect of reducing germination. Now, this is beyond the scope of this tutorial, but I can control these colors as well. And I could make the colors so that they're more representative of cold to warm temperatures. Okay, and that is also one line of code. It's very easy to do, but it's beyond what I'm going to do here today. Well, this is a picture that allows us to look at temperature effects within species, but I may be interested in focusing on what's happening between species for a given temperature. So my main interest is in comparing species. Yeah, so I can look at the species effects for each temperature. And what I'll do is I'm going to copy this code so we can see how easy this is to do. What would we do if we wanted to reverse the roles of temperature and species? Very easy. Every time I see a temp, I'm going to put a species. So here also, I'll need a species. And every time I see a species, I'm going to put a temp. So I just reverse the role. And so this way, I hope you could start to see the power of ggplot. So now I'm going to have a separate graph for each temperature. And then within each temperature, I'm going to be looking at the effect of species differences. And I can see that some species have a later germination period, et cetera, yeah, compared with others. And that pattern seems to be different for different temperatures. Yeah. So another way of looking at the data, but really it's just a question of manipulating who's on the x-axis, who is the color, and who is facet wrapped. Yeah. So it starts to give us looking at the data from very different perspectives. Great. So that's all I'm going to be doing for ggplot. So happy to take questions.
There's been a bit of discussion, Terry, about the difference between facet wrap and facet grid. I don't know if there's anything more you would add to what's already in the chat. You may not have seen the chat yet because you've been talking.
No, I haven't seen the chat. Yeah, they're very similar, right? I mean, clearly what facet grid puts a certain structure, right? It creates these rows and columns. I tend to use facet wrap because I don't know, I've just grown used to it. I think I could write a species in front in facet wrap. I'm not sure. Yeah, so I'm afraid I can't really add to the discussion. I think most of us have learned things as we need them. And so, yeah, so, yeah, okay. So,
Terry, there's another interesting question about, is it easy to make the data cumulative, the germination data cumulative, or does that take a bit more massaging?
That's a really good question. And because I've often seen data written as cumulative. And I think that's pretty easy to do, but I would have to look it up. So I could send that code. I could just make that code public or add it to the current document. Yeah. But there is a function called cum sum. Yeah. So and we could use the group by and then do a cum sum, but I have to play with it. Okay. Any other questions? Okay. Right. Oh no, there's a little bit more ggplot, and but this refers now to looking at and this is really going back to Sergey's stuff. So what Sergey decided he wanted to do was to go back to our germination 1 data, the one with 300 observations, and let's add a new column. And what that new column is going to be is the percent of germination. So what do we need to do to get the overall final percent germination? Is that we're going to need to add up these columns and then divide by seeds. And if we want it in terms of percent, we're going to have to multiply that by 100. Okay. So how do I add a new column? We've talked about this before, and I may do this up here in my data wrangling, since it is a wrangling statement. And I will say add add new columns. Now I'll do this in 2 steps. So I'll actually add 2 new columns. I'll add the sum of the germinations, yeah, and then I'll also add the percent germination. And we discussed this before, that when I add a new column, I'm going to use this function called mutate. Okay, and I will call, and I can. I'm just going to add it to the current data set. So I'm going to start with germ1. And with germ1, I will start with germ1 and I will pipe that into a mutate, which will allow me to add a new column. So mutate, and what did I decide to call it? I called it cumulative germination. So I called it cum-germ. And well, how do I do it? Well, there's a naive way that I could do this. I could just add those columns, D0 plus D3 plus D7. And I have to hope that I don't forget any of them, plus D12 plus D17 plus D24 plus D30. Okay, so this will create a new column called cum-derm. Now, I can add a column. I don't need a new mutate statement. I can put a comma after that. And before I close my mutate statement, I'm going to add a final germination percentage. And I can refer to this new variable that I've created, even though I've created it within the same statement. So that will just be my cumulative germination, yeah, divided by the total number of seeds I have, which was called seeds. And then I'll multiply that to get a percentage times 100. Okay, so now I will have created 2, and let's run it and just check to see what it's done. Whoops, D30, didn't like because it was D31. Okay, let's run that. So it sends me a prompt, so it's happy. Let's go back and have a look at germ1, and we can see that it's added 2 more columns, a column called cum-germ, which we can just double check for a few of the values to make sure that adds up to 10, and it's created this percent germination, which is 10 divided by 14. So we could check a couple of those that it's doing the right thing. Well, this one, clearly, cum-germ is 3 out of 9, that's 1/3, or 33.33%, okay? Or we can check 6 out of 8, which is 75%. So we get it, it's important to kind of make sure that the computer is doing what we think that it's doing, okay? Now, you could see that although I could do this, I could easily make a mistake. So what could I do instead? well, rather than write this, we have some clever ways. And it's just, again, it's just indicating the power of R. Yeah, you know what, I one of the problems that I have now is that I've overwritten germ 1. So now germ 1 is the same. It doesn't actually matter because I could still run this again. It will just recalculate these columns. Yeah. I'm going to copy this and just give you another bit of code to give you a feeling for how powerful. So rather than having to write every one, because sometimes I may have checked the germination maybe 10 times or 14 times. So there is a function called row sums. And I could just say, just add across. And then I just specify the first one and the last one. Okay. So nothing should change. It's going to, again, add them all up and then calculate the percentage. So I run that. It's happy, happier. It's less error prone. And it's given me, it's basically just recalculated those columns. Okay. What do I want to do with these data now? I'd like to look at them. I'd like to look at the final germination percentage. And I want to give you a few more things you can do. Yeah. So let's go back and do our data visualization. And this time we're going to look at that final germination percentage. So now my data set ggplot, I'm going to use my germ1, right? Do you remember now? I need an aesthetic. What do I want on my x-axis? Well, at this point, what do we want? We have one row per species temperature dish, yeah? So I could, for example, put species on my x-axis. Notice now that it's not that this is a character field. And so it's going to create one column essentially for each species. And Y was going to be my final germination percentage. I guess that's what I called it. Yeah. And now I'll color it by temperature. Okay, so now I've got, these are the variables of interest, species and temperature. So I want to include them to be able to differentiate them in my plot. Maybe in the first instance, I'll just look at points. Often at default. And, you know, it's a good start. I've got species here. I've got the temperatures, but it's hard to see patterns. One way that makes it easier to see patterns is instead of the points, I could use box plots. Yeah. So I'm just going to copy this code so that we still have it. And I'm going to create a different geometric object. And that is, instead of geom point, I'm going to put geom box plot. Okay. You can see now how we can go from point objects to box plots very quickly. Yeah. I want to give you a few cautions about box plots, namely that within one of these boxes, oh yes, so we can start to see patterns more readily when we use box plots, because instead of having the points, it's summarizing the distribution. But one thing I should caution you about is that each one of these boxes actually consists of exactly 5 points. Okay. So it's not, I wouldn't say it's very necessarily good as a descriptor when you only have 5 points, because a box plot actually summarizes a distribution with 5 points. Namely, this is the middle point here, the median. The edges of the boxes are the second and the fourth plot point. And the whiskers, the end of the whiskers are going to take you to the first, the lowest and the highest. unless there are big differences, like here, for example, where this fifth data point is so much bigger than the other ones, then it gets put as an outlier. Yeah. There are people who really don't like box plots for the reason that sometimes they disguise distributions. They always make distributions look unimodal, for example, when in fact they may be bimodal, or they over parameterize. Yeah. In other words, I only have 5 points, or sometimes there are only 3 points, and it will still draw a box plot. But the main message is that a box plot, as a tool for exploring your data, allows you to start to see patterns. Okay. Now, this one is a bit hard to see because everything is on one plot. So one thing we might want to do is facet this, yeah, and maybe facet it by species. So what I'll do is I'll take this and almost, yeah, we're running. I really wanted to raise one more issue. So what I might do to make things a bit easier is I'm going to put temp on the x-axis and take this and facet wrap this. Because really we're going over some of the same material, and I just want to illustrate how versatile it is. And I'm going to facet wrap that by species. So let's just have a look at that. And now we can start to again see those patterns, those germination patterns. And maybe this is clearer, okay? And how easy is it to reverse the role of temperature and species? Yeah. And again, when we reverse, so this one is, you can see that the questions that are being addressed, that we can assess here are the temperature effects within species. And again, if we want to turn that around and say, within a temperature, what are species effects, then we can just reverse the roles. Okay, so Sergey has provided you with some websites where you can go and read more about ggplot, read more about facets, read more about tidyverse, read more about themes, which we haven't talked about, but ways of making your plots more visually appealing, yeah, and controlling colors, controlling the background, etc. Okay. So it becomes a very professional. In fact, you almost need to use some of these extra themes and changing backgrounds, or reviewers or referees kind of say, oh yeah, that looks just like a standard ggplot. So you could make it as individual and as meaningful as you like. Great. Now, the last thing that I wanted to talk about, unless there are some issues. Okay. So the last thing that I wanted to mention is that there are packages that are specifically for seed ecologists. And there isn't just one, there are actually several. But I'm going to introduce one so you can get a feeling for how powerful these are. Okay, so this is the one basically that Sergey has decided to introduce. And that's a package called Germinar. So I'm going to need to load Germinar in. So I'll go back up to my load libraries. And I will say I want Germinar. So library, and it's this, if I get it right. So germination metrics is another one that we've used. That's quite interesting, but we're not going to talk about that today. So library Germinar is very specific for seed ecologists. And let's explore Germinar. Now, one of the things, like the guys who wrote Germinar, they expect the data to be what's called in a data frame. Now, when I import data using tidyverse packages, it imports it as what's called a tibble, which is like which is a data frame, but it has some enhanced structure. And if I were just to take germ1 and put it into Germinar, it would choke, okay, just because that's how the package was written to expect a data frame. So it's going to expect a certain kind of data structure. So we're going to have to change the data slightly, just the structure, in order to make it able to use in the Germinar package, okay? So I'm going to call my restructured data germ2. So basically what I'm going to do is I need to tell R, this is a data frame, species is a factor, and temperature is a factor. Okay, that's what I'm going to do. So I begin with my tibble, or my enhanced structure data frame called germ1. I'm going to pipe that. And I'm going to use my mutator function again to overwrite my columns. So mutate, I'm going to want temperature to be a factor. So how do I go from a character field to a factor? There's a function called factor. So you won't be too surprised. And I'm going to go from my character field species and turn it into a factor. Sorry, it's a lowercase factor. Okay. Now, I need to make sure that it has the structure of a data frame. And sometimes you get a warning because it's kind of, data frame is sort of an outdated, deprecated idea, because the tibble is the more enhanced data frame. So you may get a warning. No, I didn't get a warning. So let's look at germ2. What will I do? I'll use my structure function, germ2, and let's just have a look at my data. Now, it now tells me, it calls itself a data frame as opposed to germ1, which called itself a tibble and many other things. So it has only the structure of a data frame, and it's created the species is now a factor with 10 levels, and their names here are the names of the 10 levels. Temperature is a factor with 6 levels, and everything else is numeric. Okay. Great. So the function that I'm going to use is called germ summary. Germ summary is a function in Germinar. I want to understand the function. I can ask about the function by putting a question mark and writing germ summary. So this is a pre-written function that they've written in this package called, whoops. So I'll run this. And now I'm in the help tab in the lower right corner, and it tells me all about this function. It tells me the syntax, but often I find that this usage thing, and it's very standard. You know, there's something that's called the descriptive, something called usage, something called argument, something called values. All of these are very standard. What I tend to do when I'm looking at a new function is I go straight to the examples. And it's much easier to understand what the examples are looking for. So I'm going to create a new object. And what are the inputs? What are the arguments I'm going to use? There's going to be something called seed n. And what's going to go, I could actually run all of these things. And then it will be even clearer. But seed n is just the seed number. So it's the denominator for that dish. Eval name is equal to D, means we just want to, the days are the variables starting with the letter D are the ones that are the day, the relevant columns. And I need a data statement. So I can take this, and this is something that I do quite often, is I will just take the function and I'll copy it. I'll put it into my, and then it's really just a matter of substituting the data, what I know. The data I know are not called FB. My data are called germ2. My seed N, that was called seeds, that's the denominator. And my eval name is equal to D. So really that's all I needed to do. Now, I think I gave it a different name. And I called that the germination index. Okay, so let's look and see what this object is. So I've just run the code. I've created a new object called gerind, germination indices. It's also 3 variables, and now it's got 300 columns, and now it's got 16 variables. Okay, we can have a look. I'll just click on it or click view, and we can see for each temperature, species, and rep. These are variables I've created before. It's taken all of those columns, D0 through D31, and it's replaced them with new columns called GRS, GRP, MGT, MGR, blah, blah, blah. And it's basically taken those data and summarized it. And what are these summaries that it's created? Some of them you can see are repeated. So this is the number of seeds that germinated, and this is the germination percent. Okay, it's the same as the ones that we already created. These next 3 correspond to germination rate or the germination time. Okay, and so time, mean germination time, I'm not sure exactly how it's calculated, but the germination rate, if we look at it, is just 1 over the time. Yeah, and the germination speed is this same column, but it's 10 times. So these all refer to speed, where the rate, time and rate are inversely related, and these are really the same. Okay. Now, if I wanted to just double check that, let's we have some very nice code that I might just share with you. I have a function called pairs that I quite like. What is pairs? Sometimes, we want to look at plots, X versus Y, but maybe we have several X's or several Y's. So we have a multi-dimensional. We want to look at a matrix plot. Okay, people have had familiarity with matrix plots. How could I do this? Let's suppose I want to look at the speed ones and just make sure that they're all related. So I might take the mean germination time, the mean germination rate, and the speed, germination speed. Yeah. So this will create a matrix plot. Let's have a look at this, germ2. Oh, not germ2, sorry, germ indices. Okay, so this will give us our matrix plot, and we can, as you can, they've kind of duplicated. So these two are really the same plot, but just inverted. And these two are also the same plot, but just, again, reversing the X and Y axis. And this guy up here in the upper right corner is the same as the guy in the lower left corner, but just, again, with the axes reversed. Yep. So speed is, for example, here on the X-axis, rate is here on the Y-axis, and that corresponds to that plot. Okay. So this is, again, a very nice way of quickly looking at your data, quickly understanding those relationships. And here's the inverse 1 over X relationship, basically. And this is just actually multiplying by 10. Yeah. So they're really measuring the same thing, but in different ways. Okay, so that gives us the time, the rate, and the speed. The other variable of interest were this one called uncertainty and synchrony. Again, these are pretty much measuring synchrony, yeah, where you have a lot of uncertainty. That means this is large. That means that the data are not synchronous, and vice versa. Very synchronous means low uncertainty. So there's going to be, again, an inverse relationship. And again, if we wanted to look at that, and well, I encourage you to look at that, and maybe we leave that as an exercise to do a ggplot and just plot synchrony versus uncertainty. Okay. Then there are auxiliary indices, which I won't be talking about. The final thing that I would like to do is show you how we can create maybe plots for publication, or arranging plots that we create and arranging them in a way that looks very nice. Yeah, because often we create a figure and we'll have figure 2 will have 3 parts, A, B, and C. So I want to do that, and I want to introduce a new package that's again a very powerful package and with very simple associated syntax, and that's called patchwork. So I'm going to take these new variables that I have. Okay, so patchwork is for putting multiple plots on a page. I'm going to create each plot separately, and then I'm going to put them together on a page. And that will be the final thing that we do. Okay. So this is kind of really in the area of sort of communicating data and publishing our data. Okay. So let's create our plots. Now I wonder why I created this gerund too. What did I do? Gerund. Well, hope this works. Okay, so let's create our first plot. So we're going to now be using the indices from gerund. And my first plot is going to put temperature on the x-axis, the germination percent on the y-axis, and I'm going to have, I used color before. We can contrast that. We could also use fill. We can compare what that looks like instead. Yeah. I remember. So Sergey decided that he just wanted to just focus on 2 species. Why did he want to focus on 2 species? It's going to be a little bit easier to see. And it also gives us a chance to use another function from our data wrangling. So I will create this guy called down here, since I've created this over here, gerund2, which I will just focus on 2 species. That will make my graphs a little bit easier to look at. Okay. So we'll start with gerund, and I will pipe that to my subsetting function. My subsetting function is called filter. And how am I going to filter? I want to say I would want to keep all the rows where species is in, and I just need to specify what the names of my species. So my species that I want to use are CER, AARV, and FEST, ALP. Okay. So that's the end of this. So this is just going to subset the rows. So if you remember, germination index had 300 observations. let's run this one. Gerin 2 only has 60 observations. Okay. So this is the data that I'm going to plot. We'll call Gerin 2. We'll put temp on the x-axis, group on the y-axis, and we will make box plots. Yeah. And I think I was going to facet wrap by species. Now he's got a couple of other things that I'm just going to add on very quickly, because to illustrate, again, for publication, what we might want to add, we might want to, when we're not just exploring the data, but we can control the labels. I can say what I want the x-axis to say. So I can call that temperature, for example. I can control what the y-axis should say. And I could write germination %, and I could even add some slightly special characters. Yeah. Let's have a look. Okay, so this is what it looks like so far. Now, we talked a little bit about changing the background. And control, and I said we can even control the text, the size of the text. So Sergey chose something we called a classic theme, and you can see how this will affect the appearance of the graph by choosing a theme, and we're choosing also the font size. So we'll go ahead and choose 12. That and I will run this, and we can see what this looks like. Okay, so now you can see we have more, we've exercised some more control over that graph. Now I can take that graph and I can give it a name, and I'll call that BP1. Okay, now when I run it, I get nothing, but it's created an object called BP1, which is storing that graph. Okay. I'm going to create a couple more graphs. So this graph, I'm just make sure I could put a title on it, but I just keep track of what I've done. This is doing germination %. So let's create one for speed and one for synchrony. Okay. So so what am I going to do? I'll do some copy pasting. Yeah. So this will be for germination speed. And all we have to do is change the relevant variables. So what do we need to change? We'll need to change this one from germination speed. I think he's chosen mean germination rate, mean germination time. Yeah. So we'll need to change also this y-axis to mean germination time. And again, I can choose a unit, which is always a good idea. And otherwise, I'm going to keep it the same. Now let's just see what that looks like, shall we? And hope it runs. Okay. So again, I had this, and now I'll give it a name. I'll call it BP2. Why am I doing this? Well, I'm going to create these plot objects, these graphical objects, and then I'm going to arrange them on the page. Yeah. So finally, I'll look at germination synchrony. Copy what I have already, and we'll change GRP, and he's changed that to synchrony. SYN. So that was a column measuring synchrony. And I'll change my y-axis as well. So that's all. It's not much that I have to do. And that was called synchrony. I don't think there are units. At least he hasn't put units. Okay, great. Let's have a look at this one before we send it off and make it into an object. It's always a good idea to just double check. Okay, so that also works. And I'm going to, Sergey called it BP3. Create that object as well. Right. So, is everybody with me? We just have a few minutes, but it's only going to take me. Gemma, is everything okay? I can see you. Yeah? Okay. So the final thing I'm going to do is to create to rearrange these three plots. And so I'm going to create a large plot, a large graphic using this patchwork package. How do I do it? Well, it's simply a matter of telling you which, and I don't use this package very much, but I know that it's very powerful and you don't have to write much code. So if I simply write that, for example, it will choose an arrangement. So it's chosen an arrangement like this. It doesn't look particularly good, but I can specify the layout. So I'll just do a plus, plot layout. And I can say the number of rows equals 2 and by row equals false. And I look at that and it starts to produce. Now, in addition, I can put A, B, and C. I think I just need to write something called plot annotation. And again, this is all something you can find out on the patchwork package. So plot annotation tag levels equals, in this case, you can choose what kind of tags you want. And again, you're going to have to follow the, and you can see I'm now creating an A, B, and a C. Now I can see that some of these things are are too big. Although I think if I zoom it, it turns out okay. Yeah, but I can control the, if I get something that where it looks like things are overlapping and it's a bit of a mess, I can control, I have a lot of controls using this guy called base size, and I can change those. Yeah. So I'm going to leave you with that, a few ideas for exploring data, visualizing data, and creating some final graphics. And it's 11:59, so happy to take questions at this point. And I think my script is over. I think Sergey included a final note that says data, and of course I agree, data visualization is crucial to convey message. Okay, and this is only the beginning of data visualization. There's some. He included a link where you can go and explore just how much you can do using ggplot.
Thank you so much, Terry. That's been excellent, and it's amazing to see your ability to code live. I'm very envious. There are some questions popping up in the chat, so if people don't mind, maybe we'll take 2 minutes for questions and then we'll head off to the lunch break.
Great.
There's one about whether, and this maybe is a general one to everybody, whether anyone's working with Python in the R package instead of R itself. maybe one we can answer in the chat later. I personally haven't come across that yet. I don't know if you have, Terry.
No, I wish I used Python, but I don't use Python. I think people use Jupyter notebooks to be able to write both Python code and R code. And rather than using the, and the Jupyter notebook is something similar to, writing script here. One thing I will mention actually is that we've created this R code. What I encourage everyone to do is to go to a little button. Hopefully everyone has this little button here that looks like a spiral notebook, and hit compile report. And you'll see that you'll get you'll get a window that says, well, what format do you want it in? I usually choose Word. When you hit the compile button, assuming that your code is perfect, and I before I run this, I would take out my install packages, but it will produce a compiled version of what you've just done that will include your code. It will include all of your comments, and it will include the output from the code. And this becomes now your trail, basically your audit trail for showing exactly what you did and how you did it. So I encourage everyone to do that.
That's a great tip, thank you. Now I'm just going to try to share my screen. I'm not sure if we need you to stop sharing yours or not, Terry, but so everyone can see the program with any luck. Do you all see the program there? Great. All right. So we've got an hour for lunch, bit of a refresh time. And what we thought we'd do, if people are interested in talking with each other in little groups, we can set you up into breakout rooms. So what I'm going to suggest is that we take until about maybe 12:20 to go and have a break, get some lunch, and then bring it back to your computer if you would like to chat with others, and we'll put you into those breakout rooms. So yeah, a bit of free time now where we can stop recording and turn off mics and cameras and everything for a bit. And at 12:20, anyone who would like to have a chat with anyone else in the course, please come back in and we'll do that. Fantastic. So welcome everyone to the next session for the day. So we'll get stuck into the next part of the workshop, part 3, linear models. We're very lucky to have Sergey with us, and he's had an early morning start and a good coffee fix. So I won't take any time. I'm going to hand straight over to you, Sergey. And we introduced you earlier. You weren't here for that, but people have heard your background and how you came to creating this workshop. So anyway, thank you very much for joining us, and over to you, Sergey.
Thank you, Lydia. Good morning. Hello. I'm sorry. It's 5 a.m. in my time zone, so I excuse. Just anyway, thanks for having me here. Thanks for this great opportunity to teach this workshop. I guess Lydia introduced me early this morning. So I'm a plant biologist, vegetation ecologist based at the University of Regensburg in Germany. Well, actually, I came to City Ecology spontaneously. Occasionally, I wanted to do a very short workshop in Regensburg with Peter Poschlert. Then I felt in love with seeds, and here we go, 10 years later, I'm teaching a workshop on data analysis for city ecologists. Right. By no means, I'm not a statistician. I, like many of you, I like to, collect things in the field, bring them to the lab, and make them germinate, right? But I had a great opportunity to work closely with some statisticians, wonderful mathematicians, who taught me that, that you have to be very careful what statistical tests you use and how you communicate with the computer. Because basically the way you code in R is the way you ask your research questions, right? And if you ask your questions wrongly, you get wrong answers, you cannot really trust. Right, I have in my part, I'm going to cover two important issues, two important statistical tools. It's well, basically it's one. It's what we call linear regression or linear models. they belong still, of course, all of you are aware of artificial intelligence, machine learning. You know, these new methods have been taking over the classical ones, but still, you know, the standard old, the old good statistical tools like ANOVA, t-test, and linear regression still belong to the most most widely used statistical tools worldwide. And I think it is important to understand how they work and how to run them properly. Right, I have a few slides. You also have them in the Dropbox. Can you see my screen? Does it work? Yeah, right. I have also a very, how can I say, a very pragmatic, yeah, I have to say that I hate teaching, you know, online, because you don't know whether the audience see my screen. Does it work now? I hope so.
Sergey, we can see it in the presenter mode is the only thing.
Oh, okay.
I think it might be something like slideshow up the top and choosing the screen or turning off presenter mode.
Okay, but I want you to see it in the full screen mode. Now? Did
Still
you?
presenter.
Yeah, I mean, oh dear.
Amy, is this something you know how to fix? RNX, Amy. Sorry, there's a few Amy's.
No, still in presenter mode.
It should just slideshow.
Say it again. Yeah, anyway, I think we can also
Just be able to click slideshow. Is that not working? So slideshow, and then at the top of the menu bar.
And this is what I'm doing. I mean, I can see now my presentation on the screen, but apparently you see it in the presenter mode.
So at the top, when next in between animations and review.
Yeah.
And then from beginning, does that not give you, does it go, untick use presenter view, just there. On the scroll bar, go across underneath where it says monitor automatic. Go across to the right. That's it. Unclick use presenter view. Fabulous. And then from beginning.
Does it work now?
Perfect.
All right. Thank you, Amy. All right. I prepared a very short presentation. So like I said, we will be coding most of the time during the workshop, but I just wanted to give you a few theoretical things and concepts you need to understand about linear models. There is a few very useful things I find you could click on after the workshop to get more information, to learn a bit more about linear regression. So of course, the internet is full of tutorials, videos, articles, but those 5 is a very good start for absolute beginners. Well, when I teach statistics, I follow a very pragmatic approach. So I always say that science or data analysis is not really much different from playing sorting books. I guess many of you had this toy when you were younger, much younger. You know, you have a wooden box. It could be, of course, any shape, and you try to fit, you know, wooden figures of different shapes into suitable holes. And this is actually what we do as scientists, basically on a daily basis. You have your question, you have your data, and you try to find a suitable statistical tool to analyze, you know, the relationships in your data, to find out what variables might be important in your system. And yeah, and this is me doing my PhD in the Bavarian Alps 10 years ago. Right, in nature, so basically there is a very rough classification of relationships. You have linear and non-linear relationships. So today we're going to work only with this part. From the mathematical point of view, for those who are into math, linear regression is a statistical model that analyzes relationship between response variable, so usually it's noted with y, and one simple or multiple explanatory variables, x. So the difference between multiple and simple linear regression is a number of explanatory variables or predictors. We call them in the statistical language. Right, basically you have here your response variable, you have predictor or several predictors, and you have two coefficients to estimate. The computer calculates for you. Right? This important part is a random error. So there is no a perfect model. That means that there will be always a part of variability in your data your model cannot explain. Of course, by scientists, we try to understand the relationship or relationships between response and predictors as